Hello everyone, welcome to Visionary First Step YouTube channel. Today I am going to explain you the topic sexual reproduction in flowering plants. See, whenever the term sexual reproduction comes, we will try to understand the basic mode of reproduction which is present in the organisms. There are mainly two types of reproduction. One is asexual mode of reproduction, another one is sexual mode of reproduction. Now, we have studied that in our earlier classes. Asexual mode of reproduction, where vegetative part of the organism will get separated simply by fragmentation, and that fragment will grow into a new organism. But in higher plants, we observe specialized structures which help in sexual mode of reproduction, where we observe interaction of male and female gametes, and fusion of these gametes give rise to formation of zygote, and zygote develop into embryo, embryo develop into young ones. So this is the sequence how the sexually reproducing organisms are produced. Now, when you look at this slide, what do you feel actually? So this is one beautiful hibiscus flower. Now, why these flowers are produced in flowering plants? These flowers are the primary structures which help in sexual mode of reproduction in angiosperms. So what and all the whorls are present in a flower which helps in sexual mode of reproduction? Outermost whorl we call it as sepal, and inner to that petal is present. See, these two are actually accessory whorls. Now, why they are present actually? Sepals which help the flower in bud condition, so that the floral bud will not undergo any type of damage. So the primary function of the sepal is to give the protection. Petals, as we all know, here you can see such a beautiful petals are present, but why they are present? You know, it's a type of attracting the insects, or you can say pollinators. Angiosperm, they mainly require pollinators for the pollination, which will finally lead to the sexual mode of reproduction. So to attract, they have a very beautiful petals. In other words, it is also called corolla. Okay, corolla is for petals, calyx is for sepals. So these two structures in the floral whorl, they indirectly help in sexual reproduction, but directly androecium and gynoecium involve in sexual reproduction. The common name is stamen, which is the male reproductive structure. In taxonomy, we use the term androecium. Similarly, for female reproductive structure, we call it as pistil, and in taxonomy, these structures are called gynoecium. Okay, so if we observe, there are four floral whorls. In that, two are accessory whorls and two are essential whorls. Accessory whorls are sepal, and in taxonomy we use the term calyx, which is the outermost, green in color, and inner to that petal is present, which is the common word we use to represent the colorful part of the flower, and corolla is the term which commonly used in taxonomy. Now, essential whorls which mainly involve in sexual reproduction, that is stamen is the common name. Male part is also known as androecium in taxonomy. Female reproductive part is pistil. Sometimes we also use the term carpel. See, pistil is only representing female reproductive part, that is stigma, style and ovary. Later we will study in detail. Sometimes we use the term carpel in taxonomy. The only reason is flower may have more than one female reproductive structure. At that time we cannot use the term pistil, so we use the term carpel. Now gynoecium is the term used in taxonomy. Okay, again we'll just look at this flower. Just observe, this is the stalk of the flower. What we can call this stalk of the flower? In first year we studied this. This is called pedicel. Okay, so this part is pedicel. Above there is a thalamus. Here you can see this is the thalamus part, receptacle part from which floral whorls will originate. Outermost here you can see this is not calyx, okay, it is epicalyx, observed in hibiscus. Okay, next comes calyx, common name is sepal, and then corolla. Here you can see the corolla, they are all fused actually, gamopetalous condition, and here you can see the gynoecium part. This is the gynoecium part, female reproductive structure, which passes through the tubular structure, in this case of hibiscus. Here you can see actually there are 5 gynoecium, they are all fused and passing through the funnel like structure that is formed by the fusion of all the androecium. That condition we call it as monoadelphous condition. Okay, so this is how we'll study a flower. Now tell me, who is not interested in flower? Everybody likes the flower. Flower is a fascinating organ. Why? Because for any aesthetic occasions we use the flower. To congratulate someone we use the flower. To celebrate any festivals we use the flower. For daily puja we use the flower. Women's in India, they used to wear the flower daily because they have a beautiful fragrant. So flower is a fascinating organ for all the organisms, for even birds, animals, insects. They all like flower because plants, they provide certain kind of rewards when someone visits the flower, either in the form of fragrance or in the form of nectars, or in the form of beautiful colored flowers. So this is how flowers, they are the fascinating organ. Now this is again a typical flower showing different parts. Here you can see the thalamus, above that floral whorls are present, outermost is the sepal, and the large colored showy part is the petal, and male reproductive structure is androecium and female reproductive structure is gynoecium. Now coming to the main part of the topic. Here we segment this topic into 3 main parts. One is pre-fertilization event, fertilization process and post-fertilization events. Now tell me, what is fertilization actually? Fertilization is nothing but the fusion of male and female gametes. Now the process of formation of male and female gamete, we call it as pre-fertilization structures. See, for the formation of male gamete and female gamete, there should be a specific structure in the plant which help to bear the male gamete and female gamete. So initially we study the structure which helps in male gamete, nothing but the androecium, and a structure which helps in production of the egg, nothing but the gynoecium. Okay, that we will study in detail in the pre-fertilization structure and event. Event in the sense how these structures, androecium and gynoecium, they produce pollen grain and the egg. That we will study in detail. Fertilization in the sense, now once the pollen grain are produced, either by the wind current or by the insects or by the different mode of transport, how they reach the stigmatic surface and what happens on the stigmatic surface, the pollen grain, they germinate and they reaches the ovule where it fertilizes with the egg. Okay, this process we call it as double fertilization, that we will study in detail. It's a very interesting topic. That is about the fertilization. Post-fertilization in the sense, what happens after fertilization? Zygote is formed. What happens to zygote? Zygote develops into embryo. What happens to embryo? Embryo is present in the seeds. That means seed is nothing but the what? It is the fertilized ovule. Where these seeds are present? Inside the fruit. Then what is fruit? Fruit is nothing but the fertilized ovary. So this is how we study the structure of fruit, structure of seed, structure of embryo, all those things in the post-fertilization events, after fertilization. So this is the main segments of sexual reproduction of flowering plant topic. So let us start with the pre-fertilization structure and events. Hope you have a little idea about the floral primordium. You know what is leaf primordium? Part of the shoot which has the destiny to become leaf, they are called leaf primordium. Similarly, once the plant attains certain maturity, the part of the plant will decide this part will become a flower. So there are so many hormonal changes takes place in the plant body, like auxins are produced, cytokinins are produced, then some of the hormones specifically for flower production are produced. These hormones make certain changes in the plants and part of the plant will destined to become a flower, and that part we call it as floral primordium. This is very, very important term. This floral primordium later develop into a inflorescence, and this inflorescence later produces the flowers. Okay, now what and all the hormones are present, that already we studied in first PUC, plant growth and development. Mainly the auxin, cytokinin, especially cytokinin and ethylene, they play a very important role in production of flowers. Now inflorescences are formed as I told, and then they bear the flower. In flower we observe male reproductive part, that is androecium, and female reproductive part, that is gynoecium. Okay, now let us study in detail about the male reproductive structure. The common name is stamen, and inside the stamen what is present? Microsporangium. This microsporangium contains pollen grains. So this all together nothing but the male reproductive structure. Now how many of you have observed the flowers? Everybody, right? So we just observe the flower, but observe in detail how these whorls are arranged. Just observe the stamen, from where it is originating, whether it is originating from the thalamus, whether it is originating from the petal, or which type of stamens are present. They are long or short, or the lobe of the stamen is kidney shaped, or is it is elongated. So there are so many types of stamens are present. It is not the same in all the flowers, and it varies from flower to flower. Even the number of stamens varies, length of the stamen varies, shape of the stamens varies, number of pollen grains varies. So it is not same in all the flowers. Now can you tell me why it varies? It mainly depends on type of pollination, because let us consider a type of flower which is very small, inconspicuous, and it is not attracting any type of insects or pollinators for the pollination. How will it reaches its male gamete to the site of female gamete, right? So at that time it produces numerous pollen grains, and then pollen grains through the wind or water reaches the stigmatic surface, pollination takes place, right? Now how these stamens are attached to flower? They have a long and slender filament which originate either from the thalamus or from the petal. If it is originating from the petal, then the condition is called epipetalous condition. Okay, so this long slender stalk called filament and a terminal generally bilobed structure is called anther. In the next slide I will show you the picture. Here like this slender filament is present, here lobed anther is present, and filament and the anther are connected by the connective. Okay, this is the anther, this is filament. Now here there is one very important statements which usually ask in competitive exams. What is that actually? The proximal end of the filament is attached to the thalamus or the petal of the flower. Why it is given proximal, right? So it is referring to the thalamus. Okay, that is why they have given proximal end. Sometimes in competitive exam what they will do, instead of giving proximal end, they just change one word and make it distal end. So be careful while reading this statement. Proximal end of the filament attaches to the thalamus. That means in the filament this is proximal and this is distal. Suppose somebody frame the sentence like this: the filament of the anther in the distal and anther is present. Whether this statement is correct or wrong? Absolutely right, because they have already mentioned that proximal end is attached to thalamus, then anther should be present on the distal end. So be careful about this particular statement, because in one of the NEET exam they asked this. Okay, very important. The number and the length of the stamens are variable from species to species. Take different flowers, take hibiscus flower, take jasmine flower, or take any of the flower of your garden. Just observe the stamens, they are not same, they are different. Now a typical angiosperm anther is bilobed. This is very important, and they have mentioned that typical angiosperm anther lobe. That means there are chances this lobe is single lobed, there are chances it may have two or more lobes, but in common in most of the plants we observed bilobed anther. Bilobed in the sense what? Now if I say, see, this is one anther, the lower part is the filament, upper part is the anther. If you just observe that, it is like this with the two lobes and they are fused. Okay, so that is called bilobed anther. What is there inside the bilobed anther? That we will see. Okay, often longitudinal grooves run lengthwise and separating the theca. We'll see what is theca in this slide. Just observe here. See, this is one anther lobe. it's clearly observed that this is one lobe, this is another lobe, bilobed condition, or you can say dithecous condition. Here in the transverse section we can clearly observe that these are the two lobes. Now each lobe is having two chambers like this, right? So bilobed dithecous condition, each lobe is having two chambers, that is why they are called dithecous condition. Suppose in competitive exam, if they ask how many microsporangium is present, what you will say? How many are there in one anther? In one anther it is 4 microsporangium. Nothing but how many pollen sacs are present? 4 pollen sacs are present, because microsporangium develop into pollen sacs. We will try to understand words one by one. The bilobed nature of the anther is very distinct in the transverse section of the anther. So only when we take the transverse section and observe it under the dissection microscope, we'll get to know the bilobed condition. Okay, clearly. Now the anther is a 4-sided structure consisting of 4 microsporangia located at the corners, 2 in the each lobe. Here we can clearly observe the microsporangia. See, try to understand by the term microsporangia. Micro is something representing the gamete which is smaller. That is why the term microsporangia. Microsporangia is the structure inside the microspores are present. That means it is something related to male reproductive part of the flower. Now, in this anther, the microsporangium is very clear only after taking the section. Now, where they are present at the corners. If you observe the bilobed anther and take a transverse section, in the corners you observe the chambers, and inside the chamber what is present? Microsporangium is present. How many present? Each lobe consisting of 2 microsporangium. Now this microsporangium matures and develop into pollen sacs. Now why the term pollen sac? Because inside that pollen grains are produced. How they are produced, that we will study in detail in Microsporogenesis. Genesis something to related to the formation. Clear? Now the microsporangia develops further and becomes the pollen sac. There is a reason to give this statement in a different color. This question is asked once in examination. Which part will directly develop into a pollen sac actually in the male reproductive structure? That is microsporangia. Okay. They extend longitudinally all through the length of an anther and are packed with the pollen grains. So if you observe here, they are present all along. It is not confined to a one place of the anther. You cannot say it is just present at the base of the anther, or it is located at the tip of the anther, it is located only in the middle of the anther. Not like that. This microsporangium run all along the anther. Is it clear? It is present fully inside, and inside that there are specialized cells. They are called sporogenous tissue, and they develop into microspore mother cells, and these microspore mother cells are also known as pollen mother cells. And these pollen mother cells undergo meiosis, very important. Meiotically they produce the pollen tetrads, and pollen tetrad further develop into pollen grains. So I will explain that in detail. Hope this is clear. These microsporangium, they bear the pollen grains. Now just look at this diagram. This is very, very important part. What is there inside the male reproductive structure? That means if you take the transverse section of anther, what and all layers are present, and the innermost layer what is there, and what is the function of all these tissues. that we will study in detail. Structure of the microsporangium. Just observe here, this is the generalized outline of the anther, and this is one part enlarged. Here you can see the same structure, but it is in the dehisced condition. That means after maturation, wall of the anther will dehisce and help in release of the pollen grains to the outside environment. Now, so this is the young anther. Just observe, this is the young anther. When you observe this enlarged part, the outermost layer, here you can see the outermost layer. This is the outermost layer, and we call it as epidermis. Epi means outermost, dermis means layer. Next to the epidermis is the endothecium. Again, one more layer is present, a layer of cells. When you look at all these cells, they are simply a parenchymatous cells. They are compactly arranged, not tightly arranged. There is a intercellular space. Okay, so endothecium is present just below the epidermis. Please remember the sequence of arrangement of these cells, because sometimes a sequence based question is asked on this topic. The outermost is epidermis. Next to epidermis is the endothecium. Next to endothecium is the middle layer. Here there is one important aspect. Epidermis is single layered. Endothecium is again a single layered, but middle layer is multi-layered. Okay, so here you can see the middle layer with the many layer of cells, and the innermost, here you can see, so these are the tapetal cells. Tapetum. Actually, in Latin, tapetum means just a cover. Okay, so why here tapetum term is used? Because it is covering the inner pollen sac. So the term tapetum. There is one interesting aspect about this tapetum. All the cells, epidermis, endothecium, middle layer, they are all the type of cell where only one nucleus is present. But tapetum is a specialized type of cell. When the time they are formed, they have two or more nucleus. Now question comes, why they have two or more nucleus and what is their role? Epidermis, endothecium and middle layer, they mainly involve in dehiscence. Once the anther is completely dehydrated, pollen grains are fully formed and they are mature. These three layers, they absorb water from the atmosphere and they rupture and pollen grains are released. So that is the function of three layers: epidermis, endothecium and middle layer. Then what is the role of this tapetum actually? I just said tapetum in the sense in Latin word, Latin it is meaning is just a cover. So this covering cells, tapetal cells, they act as a nursing cells, or it nourishes the developing pollen grains. So it has to do multiple activities. So they have two or more nucleus which involve in helping those activities. Okay, and they have a dense cytoplasm compared to other cells. Now inner to this is the sporogenous tissue. Here you can see this part actually consisting of sporogenous tissue. What is the role of this sporogenous tissue actually? Very interesting part. Sporogenous tissue are actually dividing cells, just like a meristematic cells. These sporogenous tissue at one particular time, they divide mitotically and increase their number, and they become pollen mother cell, or you can say microspore mother cells. These microspore mother cell later they undergo meiosis and produces the pollen grains. Now the same thing is explained here. The role of epidermis, endothecium, middle layer and tapetum. Again remember the sequence properly, because sequence based questions can be expected from this topic in competitive examination. Okay, Here I just included a small explanation related to this. It is not there in NCERT, but you should know what it is. Tapetum possess dense cytoplasm and generally have more than one nucleus. To support their vital role in nourishing developing pollen by boosting the metabolic activity, increasing gene expression, and facilitating rapid synthesis and transport of nutrients like sugars and proteins essential for pollen wall formation and viability. So these are the different functions done by tapetal cells. Okay, so it is just a addition of information to tapetum, because you need to remember this tapetum, a most important aspect in studying the transverse section of anther. Okay, so can you think of how tapetal cell could become binucleate? Sometimes they will ask this question. So this is mainly because of endomitosis. Very important. Here mitosis takes place, nucleus undergo duplication, that means karyokinesis takes place, but which is not followed by the cytokinesis. If cytokinesis takes place, this will become a two cell, but it will remain as a single cell but has two or more nucleus. That is possible mainly because of endomitosis. Okay, when the anther is young, the group of compactly arranged homogeneous cells called sporogenous tissue occupies the center of each microsporangium. As we studied, in an anther, two lobes are present, corner of the lobes inside consisting of 4 chambers, and that is called microsporangium, and in the core of the microsporangium we observe a special type of cells and they are called sporogenous tissue. Why they are specialized cells? Because these sporogenous tissues, during the time of microsporogenesis, they become microspore mother cell or pollen mother cell and helps in the formation of pollen grains. Is it clear? Okay, let us move on to microsporogenesis. Microsporogenesis is very interesting process. So what happens here? This all takes place inside the anther. Okay, we studied epidermis, endothecium, middle layer, tapetum, and after reaching the center of this microsporangium, there is a group of tissue and they are called sporogenous tissue. What is the main role of this sporogenous tissue? They undergo mitosis and increases their number and become microspore mother cell, or in other words we can also say pollen mother cells, and they undergo meiosis and produces the pollen grains. Okay, so in other words you can simply say sporogenous tissue, they undergo meiosis and produces the pollen grains, in order to just have a sequence. No need to explain that sporogenous tissue undergo mitosis and increases their number and becomes microspore mother cell. You just can say that sporogenous tissue undergo meiosis and produces the microspore tetrad that separates and later become the pollen grains. Now, what would be the ploidy of the cell of the tetrad? Do you know what is ploidy? Ploidy is nothing but the number of chromosomes in a cell. Now, when the cell which is diploid undergo meiosis, how many number of chromosomes will be retained in the daughter cells? That is haploid. That means half of the number of chromosomes are retained in the microspore tetrad. So the ploidy of the microspore tetrad is haploid. As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or microspore mother cell. That means don't get confused with this sentence. How this sporogenous tissue is capable of giving rise to a pollen grain and microspore mother cell. Not like that. The meaning of the sentence is something like, this sporogenous tissue is capable of giving rise to a pollen mother cell. Each one is capable of giving rise to a pollen mother cell. Similarly, each pollen mother cell is capable of giving rise to a pollen grain. That means what? Each sporogenous tissue is capable of giving rise to a pollen grain as well. Okay, that is the meaning of this statement. The process of formation of microspores from the pollen mother cell through the meiosis is called microsporogenesis. That is the definition. What is the definition? The process of formation of pollen grain from the microspore mother cell is called microsporogenesis. The microspores, as they are formed, are arranged in the cluster of 4 cells and they are called the microspore tetrads. Now imagine, inside the microsporangium, now sporogenous tissues are present. These sporogenous tissues later become a microspore mother cells. Now microspore mother cells undergone meiosis. In the first meiotic division, they formed dyad. In the second meiotic division, they formed the tetrad. Now in these tetrads, they immediately will not undergo separation. First, in the first phase, they are together and forming a tetrad, and that we call it as microspore tetrad. As the anther matures and dehydrate, the microspores dissociate from each other and develop into pollen grains. So the pollen grains are observed only after anthers are fully matured and when it is ready to dehyse and release the pollen grains. Okay, so inside each microsporangium, several thousands of microspores are pollen grains are formed and that are released with the dehiscence of anther. Now tell me why there is a need of production of several thousands of microspores or pollen grains. Because it is a chance factor. Nobody can assure that produced every pollen grain lands on the stigmatic surface and undertake the pollination. Some of them may be lost, some of them may be carried by the insects as a source of food, or some of them will fall off. So certain will reach and produces the forms the function of fertilization. Similarly, in the case of human beings also, we all know that we produce millions of sperms to reach one egg. What is the reason? Because in that millions of sperms, most of will die in the movement or in the travel from the female reproductive tract to the egg. Similar things happens in plants also. So the number of pollen grains or the shape of the pollen grain, weight of the pollen grain and sticky nature of the pollen grain all will vary from species to species. Why? Because each and every plants are unique in their mode of pollination or fertilization process. Okay. Now let us go in deep and depth regarding the pollen grains. Here I included one picture. It is not there in your NCERT, but it is the photograph of the pollen tetrad. Just observe. It is just formed pollen tetrad. All the 4 pollen grains are together. All the 4 pollen grains are together in the tetrad form. So this is from the different species. These pollen tetrads, once they get separate, they become pollen grains. So here the pollen grain shown is not from one species, it is from the different species of plants. Now what happens to this pollen grain? Initially it is single, it has a single nucleus, that is haploid nucleus. Now this haploid nucleus pollen grain starts to develop a vacuole, and this nucleus undergo mitotic division. Very important. Haploid cell again undergo mitosis. During that time it forms the asymmetric spindle. Very, very important character here. Usually cell division, at the time of cell division, what type of spindles are formed? Symmetric spindles are formed. But here surprisingly it forms the asymmetric spindle. The reason is the next forming cells are of not same sized. They are of not same sized, because one will become a generative cell, another one will become a vegetative cell. They both have nucleus, and one is small, another one is large. In other words, there is one beautiful sentence given in your NCERT textbook. What they say: generative cell floats in the cytoplasm of the vegetative cell. See how beautifully they have given. The reason is, these vegetative cells, the nucleus which is help in formation of pollen tube. Okay, in other words, you can also say pollen tube nucleus. So which guides the pollen tube, and generative cell, it floats in the cytoplasm of the vegetative cell and reaches the female reproductive part. Okay, so that is why sentence is used. Here this picture is given in NCERT. This is the picture given in NCERT, and these three diagrams I have taken it from the different source. Hope this diagram is very clear now. Now tell me, how many cells are present in the matured pollen grain? 2 cells, one generative cell, one vegetative cell. Is it same in the case of all the plants? No. 60% of the plants, they release their pollen grains in the 2-celled stage. Remaining 40% of the plants, they release their pollen grain in the 3-celled stage. Now again a question, what is 3-celled stage? Very important. This generative cell further undergo mitotic division and form a 2 generative cell. These 2 generative cell has 2 male gametes, and which are responsible for double fertilization. One of the male gamete go and binds with the egg, and fertilization takes place. Another male gamete go and bind with the polar nuclei, and fertilization takes place. These two takes place together. That is why we use the term double fertilization. Now all the details regarding to pollen grain I explained. Once again they have given here. Pollen grains represent the male gametophyte. Please do remember, because there are so many terms here: male gamete, male gametophyte. If pollen grain is the male gametophyte, then what is male gamete? Generative cell is the male gamete. Please remember, most of the time in competitive exam, they will confuse you with the microsporangium, microspore mother cell, then pollen mother cell. Similarly, male gametophyte, male gamete. So these are the some of the terms which creates lot of ambiguity or confusions in students. Make sure that pollen grain, remember, pollen grain contains male gamete. So pollen grain will be male gametophyte. The structure which bears the gamete are called gametophytes. So the pollen grain is gametophyte. Pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter. So sometimes they will ask the size of the pollen grain also. They have given ranges here, 25 to 50 micrometers. It is in micrometers. It has a prominent two layered walls. Now we will move on to the detailed structure of the pollen grain. So far we just studied what happens to pollen grain after tetrad is formed. Now what and all the changes takes place in the pollen grain before it will become mature, that we should observe. In the last topic, while explaining the tapetum, I explained that this tapetum helps in formation of different layers of the pollen grain in the later stage. Just observe this pollen grain. This is the inner part where cytoplasm is present. There are 2 nucleus. One is tube nucleus, nothing but the nucleus of vegetative cell. Another one is the nucleus of generative cell. Now this generative cell floats in the cytoplasm of the vegetative cell. To this particular pollen grain, internally it has intine, externally it has exine. Intine is a continuous wall layer, exine is not continuous, it is interrupted. Interrupted with what? Germ pores. Why these germ pores are important? Through which pollen tubes are formed. Imagine now pollen grain in the wind reaches the stigmatic surface. Now it has to germinate the tube, then only it can reach its male gamete to the female reproductive structure. That's why germ pore is very important structure where exine is absent. For your competitive exam, all these details are very important. Intine is entire, exine is interrupted. Where it is interrupted? Where germ pores are present, their exine is interrupted. Now, what is the composition of exine and what is the composition of intine? We all know that pollen grains can be stored for several years. Do you know what is viability? Viability means it is a duration in which the pollen grains have ability to germinate and produce the pollen tube. That is viability. This viability will vary from plants to plants. In the later part, we will discuss in detail. Now, this exine made up of sporopollenin. Please remember this word, sporopollenin, which is one of the most resistant organic material now. In exam, all these will be matters a lot. Exine is made up of sporopollenin. Sporopollenin is a type of organic material. It is not inorganic. Organic material, and it can withstand high temperature and strong acids and alkali. Because of that, no enzyme that degrades sporopollenin is so far known. Illiya tanaka, that means till here nobody had discovered the type of enzyme which have capability to degrade the sporopollenin, which is present on the exine wall of the pollen grain. Now, this pollen grain exine has prominent apertures called germ pores where sporopollenin is absent. So sometimes in exam, if they ask where the sporopollenin is absent in pollen grain, your answer will be on the germ pore region. Pollen grains are well preserved as a fossils because of the presence of sporopollenin. For many years we can store them or preserve them. The only reason is because of sporopollenin, because so far there is no enzymes are identified which have ability to degrade this. The exine exhibits a fascinating array of patterns and designs. So if you observe the slides, here you can see a fascinating array, means arrangement of exines. So some of them are spiny, some of them are spiral, some of them have oval shape, some of them have depressions. So there are so many types of pollen grains are present. That is mainly due to the presence of exine. Okay, the exine exhibits a fascinating array. And why do you think the exine should be hard? Why do you think it should be hard? Because it is the layer which protects the pollen grain. And what is the function of the germ pore? Helps in the formation of pollen tube. Now coming to the inner wall. As I said, inner wall is made up of intine. Okay, inner wall is made up of intine. Now what is the composition of intine? It is made up of cellulose and pectin. Please remember, again a very important concept for competitive exam. The composition of exine is sporopollenin, which is organic, and here composition of inner wall intine, which is entire, and it is made up of cellulose and pectin. The cytoplasm of the pollen grain is surrounded by a plasma membrane. That means intine is present exterior to plasma membrane. When the pollen grain is mature, it contains two cells, the vegetative cell and generative cell. This I explained. Once the pollen grains attains its maturity, the single nucleus undergo mitosis and produces the two cells, one is generative cell, one is vegetative cell. If you compare the size, generative cells are smaller, vegetative cells are larger, and generative cells, they floats in the cytoplasm of the vegetative cell. Why? How these two unequal sized cells are produced? By the formation of asymmetric spindles. That part is very important. The vegetative cell is bigger, has abundant food reserve, and large irregularly shaped nucleus is present. So this is the characteristic feature of vegetative cell. Sometimes while framing a questions, they just change the word. Here in case of giving vegetative cell, they just change it to generative cell. You should know the exact character. If you know the purpose why they have it, then finding out the solution will become very easy. See, vegetative cell is bigger. Why? Because it has to form a pollen tube. So to form a pollen tube, it requires a large amount of cytoplasm. So they have abundant food reserve, and a large irregularly shaped nucleus is present. Actually, it helps in guiding the pollen tube from the stigmatic surface to the egg. Okay. The generative cell till micropylar region it will guide, and later on they are accepted by the filiform apparatus that we will study in pollen pistil interaction. The generative cell is small and floats in the cytoplasm of the vegetative cell. Again, this statement is very important. Most of the students, they find it very difficult to understand how the generative cell floats in the cytoplasm of the vegetative cell. Hope my explanation made you to understand this very clear. Okay, so it is spindle shaped with the dense cytoplasm and nucleus. Spindle shaped nucleus. Okay, that is the characteristic feature of generative cell. In over 60% of the angiosperm, pollen grains are shed at 2-cell stage. That means remaining 40% of the plants, they shed their pollen grains at 3-cell stage. Why this mechanism is there? Then what happens in 60% of the plants? Whether they produce 2 male gametes? Of course, yes. When the generative cell, they float in the pollen tube, at that time, when they travel half of the distance, this generative cell again undergo mitosis and produces the 2 generative cell. Otherwise, remaining 40% of the plants, what happens? Even before pollen grains are released, they become 3-celled pollen grains, where it contains 2 generative cell, which is nothing but the 2 male gametes. Okay, but what happens in 60%? This generative cell undergo mitosis in pollen tube. So the remaining species, generative cell divides mitotically to give rise to a true male gametes before pollen grains shed. Okay, so remaining 40%, before the pollen grains are shed from the anther, they form a 2 generative cells, nothing but the 2 male gametes. So pollen grains are the gametophytes and generative cell are the male gametes. That is very important. Now, pollen grains of many species cause severe allergies and bronchial afflictions in some people, often leading to a chronic respiratory disorders, asthma and bronchitis, etc. So where do we observe this? Sometimes if your house is near to any plantations like acacia, or your house is near to a place where the certain type of weeds are present like parthenium grass, they produce a pollen grains. They are very minute, so they are present in the air. When you inhale, along with the air, these pollen grains will enter your respiratory tract and they severely cause allergies. Continuous exposure of individual to these type of pollen grains may lead to asthma or bronchitis. You know this, these two are the disease related to our respiratory tract. Okay, there is a difficulty in breathing, individuals who are suffering from asthma and bronchitis. It may be mentioned that parthenium or carrot grass that came to India as a contaminant with imported wheat has become ubiquitous in occurrence and cause pollen allergy. Just now I took this example. At the time of great famine, so in the time of 1947, when we fought freedom, India was in great danger because of famine. You know, there is a lot of competition for having food. So India imported wheat from British countries, and at that time, as a contaminant, this Parthenium grass entered India, and now they are spread all over the place. You know, any exotic plants, unscientifically, they, if they are exposed to any area, there are no predators for those organisms, so they establish very well, and they cover most of the surface of the land and fertile soil. Okay, so and these carrot grass is one of the headache for farmers, because it's a very massive weed, and their pollen grains used to cause allergies in farmers or the individuals who get exposed to these pollen grains. Now, apart from this bad effect of pollen grains, now coming to the positive effects or benefits of pollen grains. See, pollen in some countries, it's very passionate that to have a pollen grains as a source of food. So they take pollen grains as a tablets, as an energy source. You know, the major diet of honeybee is pollen grain actually. They just conserve the nectars for off season, but on season when the flowers are, they take the pollen grain as a source of their food. Okay, so pollen grains are rich in nutrients. It has become fashion in recent years to use the pollen grain as a source of food supplement, and in Western countries a large number of pollen products in the form of tablets and syrups are available in the market. So these are some of the pictures. Here you can see there are some box where the pollen grains are stored, and here you can see the tablet of pollen grains. There is one sachet which contains 30 tablets. You know, in some countries, these athletes, just before their event, they used to take these pollen tablets. It is assumed that by taking pollen grains, their energy will be enhanced. So pollen consumption has been claimed to increase the performance of athletes and race horses. That's why in some countries they use the pollen grains as a great source of food. Now, once they are shed, the pollen grains have to land on the stigma surface. Now, what is the primary function of the pollen grain? It's a male gamete. It's a male gametophyte carrying male gamete. Now, it has to deliver the male gamete to the place where egg is present, where fertilization takes place. So, how long do you think the pollen grains retain viability? Already I explained you. Certain pollen grains have viability for a day. Certain pollen grains, they have viability for months. Certain pollen grains, they have viability till month. Okay, here some examples are given. Period for which pollen grains remain viable is highly variable, and some extent depends on the prevailing temperature and humidity. See, this is very important. Even seeds also impacted by temperature and humidity. In the same way, pollen grains are also impacted by varying temperature and humidity. If high temperature is there, pollen grain may lose viability early. If the favorable condition is there, the viability may extend up to some time, or if humidity is high, it will change. So these factors, environmental factors, also influence. But in some cereals such as rice and wheat, pollen grains lose viability within 30 minutes. Again, these are the some very important concepts. MCQs may be formed on these concepts, because in competitive exam they go through these type of sentences where the factual data is given. Example is cereals like rice and wheat, pollen grains have viability up to 30 minutes. As soon as the flower shows anthesis, when the flower blooms, pollen grains have viability just for 30 minutes. Before that, it has to reach the stigmatic surface and pollination should take place. Okay. And in some members of Rosaceae, Leguminosae, Solanaceae, you know Rosaceae, rose plants are present, Leguminosae like beans are present, and Solanaceae, brinjal, capsicum, tomato, all these plants maintain viability for months. For months, these pollen grains, they maintain the viability. You may have heard of storing the semens, sperms of many animals, including humans, for artificial insemination. Now, there is a reason to tell this example, how the semens are stored. You know, this process is called cryopreservation. That terminology is not given. Here they have directly given the pollen grains are stored in minus 196 degrees Celsius. That process we call it as cryopreservation. In cryopreservation, in liquid nitrogen, the temperature of the liquid nitrogen is minus 196 degrees Celsius. These male gametes like pollen grains and sperm in the semen can be stored, which later can be used in pollination, or in the case of animals for insemination. So such stored pollen can be used as a pollen banks similar to seed banks in crop breeding programs. A very, very important aspect. See, genetic resource of any plant is very important for any country. So sometimes what happens, because of over exploitation, plant may undergo complete extinction. How do you conserve the genetic resource of such plants? You can store their pollen grains in pollen banks, and whenever it is required, you can go for the tissue culture where you can produce the new plants. Or if the seeds are not viable, then you conserve the pollen grains and use the pollen grain in tissue culture and generate the new plants. So the main importance of cryopreservation is you can store these pollen grains for several years. It is related to female reproductive structure in the flower, that is gynoecium. Now, here we study in detail about the pistil, nothing but the gynoecium, and Megasporogenesis, nothing but the ovule, and embryo sac. So this concept will study in detail under the second part in pre-fertilization events. In the first part we studied the Microsporogenesis and pollen grains. Here we study Megasporogenesis in detail. Let us try to recollect some of the aspects, some of the concept already we studied in first PUC, morphology of flowering plants. So what and all we studied actually related to gynoecium part. You know that there is a difference between pistil and carpel. Even though the term is representing female reproductive structure, pistil term we use at one particular time and carpel term we use at another particular time. So what is the main difference? In general, to represent the female reproductive structure, we use the term pistil. But whenever it comes to the number of female reproductive structure present in the flower, at that time we use the term carpel. So that is how we will describe the female reproductive structure as monocarpellary, bicarpellary, tricarpellary and multicarpellary condition like this. Okay. Now the question comes, the number of carpels in a flower, whether they are fused or free. If they are fused, what type of technical terms are used? Here you can see, suppose a flower consisting of only one carpel, then we use the term monocarpellary, single carpel. Suppose more than one carpel is present in a flower, then the condition is called multicarpellary condition. Have you seen any type of flowers where it produces more than one carpel? You might have observed this, but you have not keenly observed those part. If you have observed the Michaelia champaka, there it produces many carpels, okay, and those carpels are not fused, they are all free, and that condition we call it as multicarpellary apocarpus condition. They are all free. So again there are two technical terms comes. When more carpels are present and they are all fused, then it is called syncarpus condition. Very commonly observed in citrus, where many carpels are present and they are all fused to form a single ovary. So we call it as syncarpus condition. When many carpels are present and they are all separate, then we use the term apocarpus condition. So these two are the conditions which commonly observed in some of the flowers like Michaelia champaka and citrus. In some plants it is only one ovary, then we use the term monocarpellary. Now each pistil has three parts. Now if I draw a rough diagram here, now this is the stigmatic surface. Here you can see the long style and the ovary. Okay, what is this stigma actually? What is their function? Stigma acts as a landing platform for the pollen grains. Okay, so they have a small hair-like structure which helps in trapping the pollen grains. These pollen grains, when they are land on the stigmatic surface, there are certain type of chemicals which help the pollen grain to germinate on the stigmatic surface, and germinated pollen tube now passes through the style and reaches the ovary, and inside the ovary there is a chamber. That chamber we call it as locule. Okay, so this is the ovary. Chamber inside the ovary is called locule. This long part we call it as style. Now question comes, why there is a need of having a style? Stigma, of course, it is a landing platform for the pollen grain, and it also helps in trapping the pollen grains, as well as chemical which is present in the stigmatic surface allow the pollen grains to germinate and to produce the pollen tube. Now style is just like a what we observe in human beings. We produce millions of sperms, and these sperm, they travel in the reproductive tract and then reaches the egg in the middle of the journey. That means isthmic junction. See here, style actually helps in finding out the best pollen grain having ability to germinate fast and reaches the ovule and undertake fertilization. By this way, plants can select a best quality pollen grains which has a male gamete and helps in production of their offsprings having best genetic quality. Okay, so pollen tubes which are formed fast, they reach the ovule which is present in the chamber of the ovary, that is called locule, and fertilization takes place. Here we will try to understand the parts, that is stigma, style and ovary. Along with that, I also explained their role in the fertilization. Now stigma serves as a landing platform, of course we studied. The style is the elongated slender part beneath the stigma. The style length will vary from flower to flower, okay? It is not same. In some flower style is very long, in some flower style is very short. So basal bulged part of the pistil is the ovary. Inside the ovary is the ovarian cavity, that we call it as locule. See, this locule, sometimes it is one, then we use the term unilocular condition. If it is 2, then we use the term bilocular condition, trilocular, pentalocular. So the number of locules will vary from flower to flower. The placentation is located inside the ovarian cavity. Here you can see, without placentation, these ovules cannot attach to the ovary. So there is one cushion-like structure inside the ovary which will help the ovules to attach, and that we call it as placenta. There is one definition for this. Arrangement of ovules inside the ovary is called placentation. Okay. Arising from the placenta are the megasporangia. Now this term is very important. Don't get confused. Megasporangium is different, megasporangia is different, megaspore is different. So likewise, you should be very careful while using these words. Okay. So megasporangia commonly called the ovules. Now megasporangia is nothing but what? Ovule. Why they have used the term megasporangia? Because it is plural. What is megasporangium? It is singular. Suppose ovary is containing only one ovule, then we can use the term megasporangium. Suppose ovary is having multiple ovules like this, then we use the term megasporangia. So which is representing what? Ovule. Now the number of ovules in an ovary may be 1. Okay. In which plants we observe this single ovule? The examples are wheat, paddy and mango. In exam, there is a high chance that suppose 3 or more than 3 examples are given, they take these examples and frame a type of questions like, which of the following example is the odd one? Suppose they have given 4 examples, wheat, paddy, mango and papaya. What it represents actually? One of the example is having multicarpellary condition and remaining 3 are having single ovule, single ovule is present, and here it is more than one ovule is present in the case of papaya. So that is how you can choose the odd one. Okay, so many ovules are present in papaya, watermelon and orchids. You know, in orchids they produce numerous ovules, thousands of ovules. Okay, hope this is clear. See, this is the picture. Just observe the first one. So this is the female reproductive structure of the hibiscus flower. Now if you observe here, this is stigma. How many are there actually? 1, 2, 3, 4 and 5. What they represent actually? There are 5 carpels. Now they are all fused here. Here they are all fused. Now tell me, whatever we are seeing here, is it a style? Actually, this style part, it is covered by a monoadelphous condition in real flower, but here they separated it. So this is the long style and here comes the ovary. This is the thalamus on which ovary is located. Now here what type of terminology we can use? It is multicarpellary syncarpous condition. Okay. So this is the again one more example, multicarpellary syncarpous pistil of papaver. In second year you study this example one more time, papaver somniferum. Okay, so this is one plant. Here you can see the stigma and it is a syncarpous ovary. Again one more example for syncarpous. So in exam you should remember hibiscus is also an example for syncarpous ovary and papaver is also an example for syncarpous ovary. Now if you observe this one, this is actually a ovary of Michaelia champaka flower. Here you can see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, like that, so many ovaries, and they are all not fused, they are all free. So which type of technical term we use here? It is a apocarpus condition, multicarpellary apocarpus condition. Now this is one ovule. This we call it as anatropous ovule. There are many types, orthotropous, campylotropous. So here one type is given, that is anatropous ovule. The naming is mainly based on position of micropyle and chalaza with respect to funicle. Okay, so here the ovule is completely bent like this, and micropylar region is near to the funicle, so we use the term anatropous ovule. So let us study this anatropous ovule in detail. Let me explain this particular anatropous ovule by taking this diagram in the next page. Megasporogenesis or ovule. Just observe this picture, such a wonderful picture. You can see all the details. See this oval shaped body, this is the body of the ovule. This is attached to the placenta with the help of stalk, and that we call it as funicle. So stalk of the ovule is called funicle. Now funicle attaches to the body of the ovule with the help of hilum. Now this hilum is the junction between funicle and ovule. There is one strong supportive structure here. This is called raphe. Here not mentioned. Actually this strong supportive structure is raphe. And if you see the body of the ovule, here there are two layers. This is the integument. Okay, integument. This integument is interrupted in the two regions. One is at the extreme end, another here in the micropylar region, it is interrupted. So this micropyle is the region where pollen tube enters the megasporogeneum and releases the male gametes. Opposite to this we call it as chalazal end. Very, very important. Chalazal end represents the base of the ovule. Okay. Now inside, if you observe, there are so many cells. What are those actually? Nucellus. This nucellar cells act as a nursing cells. Again, they nourishes the embryo sac. Here there will be a sporogenous tissue again. They undergo mitotic division and form a megaspore mother cell. Now megaspore mother cell undergo meiosis and form a 4 cells. Out of the 4 cells, 3 will degenerate, and the remaining one, which is nearer to the micropylar region, will starts to grow and become the embryo sac in the later part of the development. So that we will study in detail in the next part. So here same thing is explained. Let us go with one by one. The ovule is small structure attaches to the placenta by means of stalk called funicle. So very important part because in competitive exam you may get many questions based on statement based. So thoroughly understand each and every line from this. The body of the ovule fuses with the funicle in the region called hilum. So body of the ovule fuses with the funicle. This part is nothing but the hilum. Okay. Now each ovule has 1 or 2 protective envelopes called integuments. Do you know what happens to this integuments in the after fertilization, they become seed coats. Okay, so integuments encircle the nucellus except at the tip where the small opening called micropylar region. So here if you observe, integument is present entirely, but it is interrupted in the micropylar region. Opposite to micropylar end is the chalaza, representing the basal part of the ovule. This is very, very important. What it represents, chalaza? Chalaza represents the basal part of the ovule. Enclosed within the integument is a mass of cell called the nucellus. Cells of the nucellus have abundant reserve food material. Why it has abundant reserve food material? Because it needs to nourish the developing embryo sac. Now located in the nucellus is the embryo sac or female gametophyte. So one more name for embryo sac is female gametophyte. An ovule generally has a single embryo sac formed from the megaspore. This is very important. This is called monosporic development. Now coming to the topic Megasporogenesis. Genesis, something related to formation. Megasporogenesis means the formation of megaspore. It's a process. How megaspore is formed? See, previously I have shown you one picture, the structure of anatropous ovule. Inside I have shown you the region where nucellus is present. Now some of the nucellar cells, they enlarge and becomes the sporogenous tissue, and this sporogenous tissue undergo mitosis and produces the megaspore mother cell. Now megaspore mother cell undergo meiosis and produces the 4 haploid cells. In that 3 will degenerate and 1 which is nearer to the micropylar region develop and form a embryo sac. That is what explained in this. Okay, the process of formation of megaspore from the megaspore mother cell is called Megasporogenesis. Ovules generally differentiate a single megaspore mother cell in the micropylar region of the nucellus. So one of the cell will retain and other 3 will be degenerated. It is a large cell containing dense cytoplasm and prominent nucleus. The megaspore mother cell undergo meiotic divisions. And what is the importance of megaspore mother cell to undergoing meiosis? Why it should undergo meiosis? In order to maintain the ploidy, because when gametes are formed, they should be haploid. Only when 2 haploid gametes fuse, they form a diploid zygote. Otherwise what happens? You just imagine there are 2 gametes, they are not haploid, they are diploid. When they fuse, what happens? The offspring will be tetraploid, which we commonly not observe in the nature. So in order to maintain the ploidy, it has to undergo the meiosis, so the gametes will become haploid. Meiosis results in the production of 4 megaspores. Here also 4 megaspores are formed just like in microsporogenesis, but in microsporogenesis all the 4 microspores are retained, but here it is different. When the 4 megaspores are formed, 3 will degenerate and 1 will be retained. Now, in a majority of the flowering plants, one of the megaspore is functional while the other 3 will degenerate. So if you observe here, in a majority of the flowering plants they have used. That means not in all the plants, all the 4 will degenerate, but in most of the case 3 will degenerate and 1 will become the functional megaspore. Only the functional megaspore develops into a female gametophyte. Another name for that is embryo sac. So this method of embryo sac formation from the single megaspore term monosporic development, very, very important. So one of the characteristic feature of angiosperms, monosporic development. What will be the ploidy of the cells of the nucellus? Now tell me, what is the ploidy of the nucellus? Of course, it is a part of the plant body. It is diploid because it has not undergone any type of meiosis. Megaspore mother cell, it must be diploid because only when it is diploid, when it undergo meiosis, it forms the haploid cells. So the functional megaspore is, of course, it is haploid, and the female gametophyte is also haploid. Simply by giving this type of question, he is giving the hint that there is a high chance in the competitive exam they ask questions on the ploidy of the cells. Okay, hope this concept is clear for you. Now, formation of the embryo sac. Again, one of the most important topic. Let me take the picture for better understanding. From this, it is very easy for me to explain the Megasporogenesis. Just observe the first picture. So this is the megaspore. This is one ovule. Here you can see the outer layer is nucellus. Inside there is one megaspore mother cell. Okay, this megaspore mother cell undergo meiosis. In the first meiotic division, it forms two cells, that is called megaspore dyad. Here you can see this is one cell and this is another cell. So this is the meiotic division first. In the meiosis second, it forms four cells. Now the cell which is nearer to the micropylar region will be retained and other three cells will undergo degeneration. Now this is the cell. Here we should start with the single cell. Okay, now this is the cell which is present nearer to the micropylar region. Now one nucleus which is present inside this embryo sac is actually a haploid nucleus. Now this haploid nucleus undergo first mitotic division. Remember, mitotic division, and form the two nucleus. Okay, now again it undergo mitosis and produces four nucleus, and half of the nucleus they move towards one polar region and another half will move towards the another polar region. Now subsequent mitotic division forms four nucleus at one polar region, another four nucleus at the another polar region. So total, how many nucleus are present? 8 nucleus is present. How many mitosis occurred? 3 times mitosis occurred. Okay, so which type of mitosis? Endomitosis. Because mitosis, when it takes place, when one nucleus becomes two nucleus, immediately it is not followed by the cytokinesis. So we can say it is endomitosis. Now once the 8 nucleus is formed, the cell nucleus starts to form a cell wall around it. The cell which is nearer to the chalazal region will become antipodal cells. So they form 3 antipodal cells. This is nearer to chalazal region. Now the part which is nearer to the micropylar region will form 2 synergids and 1 egg cell. Okay, now these 2 synergids, they act as a cell which provide nourishment to growing egg. Okay, if you see here in the micropylar region, they just flipped it. Okay, here they have given micropylar region, now here they are mentioning micropylar region. Finger-like projections are present and they are called filiform apparatus. They play very important role. When the pollen tube reaches the micropylar region, male gametes, how many male gametes are present? 2 male gametes are present. They have to be guided properly towards the egg and the polar nuclei. that guiding is done by the filiform apparatus which is present in the micropylar region. Now, male nucleus, male gametes, one will fuse with the egg, another will fuse with the polar nuclei. Now, if you look at this complete embryo sac, what you can see, there are 3 cells in the chalazal end and they are called antipodal cells. Their functions are not clear. And here if you see a very large cell, here it has 2 nucleus. The name of the cell is called primary endosperm cell, or you can say polar nuclei. They are diploid. Here antipodal cells are haploid. All the 3 cells are haploid. Polar nuclei is diploid. Synergid is haploid. And what about the egg cell? It is also haploid. Now when you look at the complete embryo sac, how many cells are present? There are 7 cells. How many nucleus is present? 8 nucleus is present. There is one important aspect you need to remember here. Once the male nucleus fuses with the egg will be fertilized and develop into embryo. Now the ploidy of the embryo will be diploid. But there is one more male nucleus. When it fuses with the polar nuclei is already diploid. Diploid plus haploid, it will form a triploid structure that will become an endosperm. So whenever the question comes, what is the ploidy of the endosperm? It is triploid. What is the ploidy of the polar nuclei? It is diploid. So you can expect many questions which is related to ploidy of the cells from this concept. Okay. So same thing is explained here. One more thing I want to make clear. Sometimes they ask questions like what are the egg apparatus? Egg apparatus means egg cell and 2 synergids. Together we consider it as a egg apparatus. Okay. So this is about the pistil, Megasporogenesis and embryo sac. Hope this concept is clear for you, because first concept, which is stamen, Microsporogenesis and pollen grain, and the second concept, pistil, Megasporogenesis and the embryo sac, is the basic concept to study all the aspects related to this topic. Hope these 2 concepts are very clear for you. So in this process, in microsporogenesis and megasporogenesis, plant was able to produce functional male gamete and functional female gamete. Now the question comes, how these 2 gametes are fused? Because what we studied in lower classes is, in primitive plants, gametes are non-motile and sometimes they are motile. When they are released into the environment, like in water medium, with the help of current of water, these 2 gametes will fuse and fertilization will take place. Now this is the advanced plant. How these 2 gametes are come in contact and how they are undergo fertilization is the very interesting concept. Here, pollination is the process through which male gamete can reach the female gamete. Now what is the definition of pollination? It's very simple. Transfer of pollen from anther to the stigma of the flower is called pollination. As simple as it is. Now how this pollination will be achieved? There are different types of plants, they adapted for different mode of pollination. It is not same in all the plants. If you observe in nature, certain plants they produce beautiful flowers. Certain plants they produce a very small flowers. Some plants they produce some smelly flowers. Some plants they produce fragrant flowers. Some they produce nectars. By all these different adaptation is there, because in the course of evolution these plants got evolved for different types of pollination. Some plants depend on insects, some plants depend on wind, some plants depend on water, some plants depends on birds, some plants depends on animals. So in some plants the pollination is done by human beings. You know, vanilla is the best example for that. So same thing we'll study in detail here. First we will study the types of pollination, and then we will study what and all are the biotic type of pollinations and abiotic medium where pollination takes place. Okay, so let us go with the kinds of pollination. Autogamy, geitonogamy and xenogamy are the three types. Let us go in detail with the autogamy. Here name itself indicates autogamy means what? The pollen grain of the same flower come in contact with the stigma of the same flower and pollination takes place. So the definition is, in this type, pollination is achieved within the same flower. Transfer of pollen grains from anther to the stigma of the same flower is called autogamy. Very commonly we observe this. If the flower is bisexual, then we can say autogamy is very common. But there is a special type here. In autogamy, we observe chasmogamous flowers and cleistogamous flowers. Okay, in simple words, if I draw a diagram here, suppose this is one plant producing a flower. Here this flower is having androecium as well as gynoecium. What happens? Pollen grains which are released from this will deposit on the stigmatic surface and pollination takes place. And this we call it as autogamy. This is just a definition. Now observe what happens continuously when the pollen grain of the same flower fall on the stigmatic surface and pollination takes place, fertilization takes place, produced offsprings are which is exactly similar to their parents. There is a very little chance that they will have variation. Okay, so this is nothing but the self-pollination. Chasmogamous flower means flower will open and stigma will be get exposed, and on this exposed stigma the pollen grains of the same flower will dust on and they germinate and produces the pollen tube, and then fertilization takes place. Then what is cleistogamous flower? It's a type of flower which is completely closed, will never open. Okay, so the pollen grains make sure that 100% the pollen grain of the same flower will pollinate the same that means stigma, that means they are deposited on the stigma. There is no chance that cross pollination may occur. In chasmogamous flower there is a chance the cross pollination may occur, but in cleistogamous there is a 100% sure that the pollen of the same flower will be transferred to the stigma. Now what and all the examples are there? If you observe in viola, the common name is common pansy, oxalis and commelina. These are the three types of plants where you observe chasmogamous flowers, that means completely opened. Okay, and cleistogamous flowers are observed in groundnut plant. Hope you have seen the groundnut plant. The flowers are produced in the soil actually. Pollination confers inside and it sets the seed and the fruit. Okay, here they have not mentioned the example. Only for chasmogamous flower they have mentioned the example. But remember, for cleistogamous flower example is groundnut. So in such flowers the anther and stigma lie close to each other. When anther dehisces in the flower, pollen grains come in contact with the stigma to affect the pollination. Thus, cleistogamous flowers are invariably autogamous. This is very important. Invariably autogamous, as there is no chance of occurring pollen landing on the stigma. That means no chance of cross pollination. Cross pollination is avoided here. Self pollination is encouraged. So cleistogamous flowers produce assured seed set even in the absence of pollinators. These plants are not dependent on the pollinators. They know that the viable pollen grains produced in this plant will 100% make sure that pollination will take place. So do you think that cleistogamy is advantageous or disadvantageous to the plant? If it is advantageous, why? If it is disadvantageous, for that also you should give a reason why. So let me tell you why it is advantageous. One thing, there is no dependency on pollinators. Second thing, there is a high chance of pollination and fertilization. And plant make sure that its next offsprings will be produced. What is the disadvantage? There are some disadvantages. The thing is, produced all the offsprings are exactly identical to their parents. So in the course of evolution, this is not encouraged actually. Cross pollination lead to variation lead to the type of offsprings which are more adaptable to the environment. So that is what every organisms expected. So this type may not be present in most of the organisms, but observed in some of the plants like groundnut. Now gynoecum, this is very interesting. What is gynoecum? It is somewhat like self-pollination only. Here transfer of pollen takes place from anther to the stigma of the different flower of the same plant. So let us say that here there is one plant which is producing many flower like this. Here there is one flower, here another flower, okay. Now transfer of pollen takes place from anther to the stigma of the another flower of the same plant. This is gynoecum. Now tell me, this gynoecum makes what difference compared to autogamy? They are all almost same, because genetically offsprings are exactly similar to their parents, but this is cross pollination similar to self pollination. Okay, so transfer of pollen grains from anther to the stigma of the another flower of the same plant. Although gynoecum is functionally cross pollination involving pollinating agents, genetically similar to autogamy. So whenever this type of statements are observed, you can expect assertion and reason type of questions from this. Suppose assertion is given like this: gynoecum is similar to autogamy. Okay, that is the assertion. Reason what they will give: even though it is functionally cross pollination involving a pollinating agents, genetically it is similar to autogamy. If they have given a reason, then assertion and reason both are correct, and reason is the correct explanation of assertion. Sometimes they will change the reason. At that time, please make sure that gynoecum is similar to autogamy for one reason, that is transfer of pollen takes place from anther to the stigma of the different flower of the same plant. That means genetic constitution is same. It is like self pollination, but functionally it is cross pollination, requires the pollinating agents. Now coming to xenogamy. This is interesting type of pollination. What happens here? Transfer of pollen grain takes place from anther to the stigma of the different flower of the same species. It is not of the same plant, of the same species. That means here there is one flowering plant. Here there is one more flowering plant. They all starts to bloom the flower. Now pollen grain from one flower with the help of pollinating agents reaches the another plant of the same species and pollination takes place. Please do remember there are certain chemicals on the stigmatic surface which have the ability to recognize the pollen grain of the same species and pollen grain of the different species. Okay, so let us consider these two are the same species of plants and at the same time they shown a flowering. Now what happens in the true cross pollination, that is in xenogamy? This is plant A, this is plant B. Pollen from the anthers are transferred to the stigma of the another flower of the different plant, but they belong to same species. Okay, so this is only a type of pollination which during pollination brings genetically different types of pollen grains to the stigma. So here offspring produced will have a slight variation, not exactly similar to their parents. They will have a variation. Why there is a variation? Because it is a true cross pollination. Okay. Now the next concept is agents of pollination. In order to transfer the pollen grain from anther to the stigma, either we require wind or we require water or pollinating agents like insects, snails, ants, birds, animals, etc. Okay, so they are acting as agents of pollination. Plants use two abiotic factors like wind and water and one biotic factor, that is animals, to achieve the pollination. Pollination is a very beautiful process. In early morning, you just visit your garden and observe the activity of insects. Suppose if you observe butterflies, they specifically visit certain plants and they actually they take reward from the flowers, they take the nectars. At the same time, when they visit different flowers, they come up with the pollination. Okay, similarly the honeybees, they also visit different flowers to achieve the pollination, but their primary intention is to get the reward. Who will give the reward? Flowers. In turn, what they will get? They will get the pollination. Okay, majority of the plants use biotic agents for pollination. Majority of the plants, we observe the pollination takes place with the help of insects or birds or animals. Now only a small portion of the plant use abiotic agents, either by water pollination or by wind pollination. Pollen grains coming in contact with the stigma is a chance factor in both wind and water pollination. So if you observe a type of plant which is showing pollination with the help of wind and water, they produce numerous pollen grains and flowers are very small. Do you think why this adaptation is there? Because if the pollen grain are carried by the wind, suppose one flowering plant is here, another flowering plant is here, and wind direction in this, then what happens? Produce all the pollen grains will be just wasted. So what happens? They produce numerous pollen grains, thousands of pollen grains. If in case the wind direction in this, here there is one plant which gets pollinated, so it's a chance factor. But in the case of insect pollination or animal pollination, if the flower has a reward, in the early morning, these insects or butterflies, they visit each and every flower to collect the reward. There is a high chance it undergoes the pollination and fertilization. So that's why whenever plant is dependent on abiotic factor like water and wind, then they need to produce a large number of pollen grains and very small flowers, because they are, if there is no insect pollination, there is no need to produce a colorful petals or any type of rewards. Okay, to compensate for this uncertainties and associated loss of pollen grains, the flower produce enormous amount of pollen when compared to the number of ovules available for pollination. Usually in abiotic pollinating flowers, one ovary will contain one ovule. Even though they have one ovule, they produce numerous pollen grains. The reason is there is a high chance that these pollen grains can be get wasted. Okay. So pollination by wind is more common amongst abiotic pollinations. Wind pollination also requires that pollen grains are light. So these are the very important characters. See, in examination, what type of questions may appear from this? They will give the question like this: select the incorrect statement with respect to wind pollination. In that, they include certain important aspects like what and all are the characteristics of wind pollinated flower. One thing, pollen grains are light. Why there is a need of producing light pollen grains? Because it has to be carried by the wind. If they are heavy, then it is not possible. That is why the pollen grain produced are lightweight. Second thing, they are non-sticky. Why they are non-sticky? There is no reason to stick on to the body of pollinators. That is why they are non-sticky. Okay, then they often possess well exposed stamens. Well exposed stamens help in waving in the wind, and pollen grains are easily released. Then, and large, often feathery stigma. See, feathery stigma helps in trapping the pollen grains. Imagine there is one strong wind current, thousands of pollen grains are released to the environment. Now the stigma, having a small hair-like structures, can trap these pollen grains easily. If there is no such things, then the pollen grain deposit on the stigmatic surface, chance of deposition will be very, very less. Okay, so wind pollinated flowers often have single ovule. This is one more character in each ovary, and numerous flowers packed into a inflorescence. Numerous flowers are packed into a compact inflorescence. Why? Because if flowers are very close to each other, there is a high chance they may get pollination from the wind current. Okay, so a familiar example is corn cob. So in textbook they have given one diagram, and the term which is used here, corn cob, is slightly different. Okay, the inflorescence of maize plant, where small flowers are produced, it's correct, male inflorescence, and female flowers are produced in corn cob, that is different. So here don't get confused. Okay, feathery inflorescence in maize plant is produced at the top. So here actually what they have given, corn cob is actually a group of stigma and style which is coming out of the corn cob. It is not the inflorescence. Okay, so wind pollination is quite common in grasses. So wherever you see monocot plants or grass, not monocot, in grasses, which is very common, wind pollination. Now next one is pollination by water. Pollination by water. See, abiotic factor like wind, they play some more role in pollination. But when it comes to water pollination, very limited genera of plants, they show pollination by water. But just remember the previous topics like in morphology of flowering plants as well as in plant kingdom. Especially in plant kingdom, we studied that algae, bryophytes, gymnosperms, they mainly dependent on water, and their distribution is also restricted to confined area, very small area, because of the complete dependency of plant on the water for fertilization. But just observe the flowering plants. Now the scenario get completely changed, because these flowering plants, they are concentrating more on the pollinators, biotic pollinators, not on the abiotic component to act as a medium for pollination. So that is why only 30 genera of plants, they are confined to water pollination. So how this water pollination takes place? See, don't get confused. Hydrophytic plants, they grow in water, on the surface of the water sometimes they are completely submerged. The plants which are growing on the water surface, that means they are floating on the water surface because of buoyancy, they produces the flowers and which are colorful, and they are pollinated by the insects. Okay, here water is not acting as a medium to conduct the pollination, but some plants, they are completely submerged and they require water current to reach their male gametes to the female gametes. Let us see how this water pollination takes place and how plants are evolved for this mechanism. Now pollination by water is quite rare. As I said, it is found only in 30 genera of mostly monocotyledons. Very important, mostly monocotyledons. Okay, so this may be a very important key point for competitive exam. 30 genera and mostly monocotyledons, because what they will do, they just twist the sentence like this: pollination by water is quite rare in flowering plants and it is limited to about 30 genera, mostly dicotyledons. Like this they will change the sentence while framing the question. So you should be very careful that most of the water pollinated plants, they are monocotyledons. As against this, you would recall that water is a regular mode of transport in lower plants like algae, bryophytes and gymnosperms. So that is why their distribution is restricted to a small geographical area. The reason is, if there is no water, there is no fertilization of gametes. Some examples of water pollinated plants are Balisneria, Hydrilla. So these two are freshwater examples, okay, plants which grow in the fresh water, Balisneria and Hydrilla. I will show you the picture in the next slide. And several marine seagrass such as Zostera. So Zostera is one more example which grow in salt water. Okay. Now, not all aquatic plants use water for pollination, as I explained. In the case of water hyacinth and water lily, it's a different scenario. They have a buoyancy, they float on the surface of water, and pollination takes place by insects. Now observe here, this is Vallisneria, very commonly observed in fish tank. Okay, so this plant is grown in the fish tank so that it will be a artificial aquatic medium for the fish in the fish tank. Here what happens, male flowers are completely submerged and female flowers, they reach the surface. So these male flowers, when they release the pollen grains passively reaches the surface of water, and through the water current these male gametes will reaches the stigma and pollination takes place. I will show you certain beautiful adaptations which are observed in Vallisneria at the time of pollination. This is hydrilla and this is seagrass, that we call it as zostera. Okay. Now just observe the pictures. This is water hyacinth. This plant is very important plant to remember in its ecological impact. The reason is this plant mainly undergo reproduction by asexual method, by forming an offset. So whenever this plant is introduced to a new niche like a freshwater lake, within very short interval of time they spread to all the area and cause trouble. They are also known as terror of Bengal. If you look at the beautiful flowers, they are very beautiful because they can attract the insects for pollination. Even though they are hydrophytic plants, they attract insect for pollination. They are not pollinated by the water. Here if you observe this plant, hope you all know this plant, right? This is lotus plant. So here we can also call it as water lily. Flowers are so beautiful which attracts lot of insects for pollination. Plant is hydrophytic. Okay, so those two are the exceptions. Now here if you come and observe, this is the Vallisneria. Just observe the female flowers, how beautiful they are. Look at their stalk of the flower. It is like a spring. Why? Sometimes what happens, surface of the water will go down. Sometimes because of rush of water level will increase. But the intention of the flower is to reach the surface of water in order to get pollinated by the pollen grains. So this spiral arrangement of floral stalk help the flower to adjust their height according to the level of water, and this flower which is completely submerged is the male flower, and that produces the pollen grain. Here a slight water surface they have mentioned, and on that so many pollen grains are there. Those pollen grains with the help of water current reaches the surface of the flower, female flower, and pollination takes place. Now this is seagrass. Slight difference. See, all these concepts are very important for competitive exam. See, in the case of Vallisneria, female flower reaches the surface, but in the case of seagrass, in Zostera, male and female flowers both are completely submerged. Okay, now pollen grains, they are released in the water, not on to the surface. They are released in the water, and with the help of water current, pollination takes place. Now one thing is very important here, how these pollen grains do not undergo wet. They have a special mucilaginous coating that protect the pollen grain not undergoing wetting. Okay, and then when they reach the female flower, pollination takes place. So when you look at these two plants, whether they are unisexual flowers or bisexual flowers, there may be a chance that they will ask a conceptual based question on this. See, they produce male flower as well as female flower. So the plants are unisexual plants, but it produces flowers, they are unisexual, but plants are monoecious. Okay, so this concept is very, very important. You should have a clear idea whether they are monoecious or dioecious, as well as they are unisexual or bisexual. Okay, so same thing is explained here, how the pollination takes place. That is what I explained with the help of this diagram. Now this is very, very interesting part, how the pollination takes place with the help of insects. So far we studied the abiotic factor which is responsible for pollination. Here onwards we will study how the biotic factor they involve in pollination, because majority of the plants they depend on biotic factor for pollination. Just observe, here I included the slide where one beetle is pollinating the flower. Here there is one more picture where one wasp is pollinating the flower. Here one flies. Here you can see wherever there is dirty things or fecal material, you observe these flies. These flies also sometimes they undertake pollination. Here you can see a beautiful butterfly is pollinating. Here honeybees, you know the importance of honeybees. Honeybees, they are the major pollinators. Without honeybees, we cannot imagine our life. The earth is completely dependent on these honeybees. Whatever the food we are taking today, it is all because of their effort. Okay, so honeybees, they every morning they visit the flower and pollinate it. And here you can see an ant undertaking pollination. Okay, so majority of the flowering plants use a range of animals as pollinating agents. Under that, bees, butterflies, flies, beetles, wasp, ant, moth, birds, especially sunbirds and hummingbirds, and bats are common pollinating agents. In the next slide, I will show you other pictures also. Okay, among the animals, insects, particularly bees, are the dominant biotic pollinating agents. So the bees, they play very important role. Now just observe this. Here one picture where you can observe a large animal pollinating the flower. So it's a mammal, okay, lemur. It is called lemur. It's a primate which undertake pollination sometimes. Then arboreal tree dwelling rodents. Here you can see the rodents. They are pollinating this flower. Okay, and sometimes lizards like zecco lizards and garden lizard. Garden lizards are very common, but observe this zecco lizard. Okay, it's a zecco lizard that also sometimes undertake pollination. So they are all expecting floral reward. What is the floral reward? See, if nothing is there, why should they come in contact with the flower? Whenever they come in contact with the flower, they should get something in return of that. So that's why they are visiting the flower. The beautiful petals are actually attracting these organisms towards it, and the sweet nectar produced by the flower help them to have some amount of nutrition from the flower. So because of that rewards, these organisms will visit the flower and conduct the pollination. Okay, often flowers of animal pollinated plants are specifically adapted for particular species of animals. So it is all evolutionary aspect. All the flowers are not visited by all the insects or all the animals. Here there is a specific interaction between the particular plant and a particular animal. Over a course of evolution, these two organisms, they coexisted. One will give the reward and another one will help in pollination. So this is how we observe coexistence of animals and plants. Now, majority of the insect pollinated flowers are large, colorful, fragrant and rich in nectar. I said this is mainly because to attract the insects. Then when the flowers are small, a number of flowers are clustered into inflorescence to make them conspicuous. You just observe this picture, okay? You will have some type of idea here. See, this is the flower of the grass. What you can observe from this picture, flowers are very small and they are not attractive. You can say it is inconspicuous, but they become conspicuous. You can see that flower only because they all clustered compactly on the inflorescence, so that they can be appeared. But plants usually they do not dependent on the insects for pollination. In grass it is mainly because of wind, but there are some plants where they produce small flowers, but they are all clustered only to attract the insects. So when small flowers together, it looks like a large flower and they can attract insects easily, and cross-pollination takes place with the help of insects. Animals are attracted to flowers by color and fragrance. You know that whenever in the early morning, if you visit your garden, flowers will have a beautiful fragrance. Why they have fragrance? We all are stimulated when we have a beautiful fragrance. Similarly, even insects and birds also get attracted when they have a very pleasant fragrance. Now, the flowers pollinated by flies and beetles secrete foul odor. You know that whenever there is a excretory material, whenever there is a carcass, these flies are attracted. Why? Because that pungent smell or that odor which is coming out of that decaying material attracts these flies. So in order to mimic that, there are certain plants, they produce the flower which produces a foul smell, so that flies can be attracted to that. But the intention of the plant is to have pollination with the help of those flies. Okay, to sustain animals visit, the flowers have to provide a reward to the animals in the form of nectars or in the form of pollen grains. Okay, so those are the floral rewards. See, there are some organisms, they are called pollen robbers. See, what they will do, they just rob the pollen grains or nectar from the flower, but they do not undertake pollination. Okay, so this type of organisms also exist in the nature. For harvesting the rewards from the flower of the animals visitor come in contact with the anther and the stigma. The body of the animal gets coated of pollen grains, which are generally sticky and animal pollinated flowers. So one thing we need to remember, when we are studying the abiotic factor like wind pollination, there we studied that pollen grains are non-sticky and lightweight, but here it is opposite. Here pollen grains are sticky and heavy. So whenever these insects they enter the flower, so usually their back surface has small hair-like structure. All these pollen grains are dusted on their body surface and it is sticky. That's why pollen grains can easily stick on to their surface. When they visit another flower, so these pollen grains come in contact with the stigma and pollination takes place. So when the animal carrying pollen on its body come in contact with the stigma, it brings about the pollination. Here you can see hummingbird which is taking nectar from the flower. You can see this in your garden every day. Hummingbirds early in the morning, they visit every flower and collect the nectar from it, because very little amount of nectar will be available in the flower. And this is the bat. Usually in the night time, bats they undertake pollination. Okay, so these plants are adapted in such a way that they bloom the flower, or anthesis of the flower takes place in the night. Now let me show you one wonderful picture here. Can you see here there are people standing over and taking pictures? Look at the flower size. Amazing, right? Why? Because this is Amorphophilus. In different language, we call it in a different name. In Hindi, it is called Zamin Kand. In Kannada, it is called Suvarnagadde. Like that, we call in different names, but scientific name is Amorphophilus. So, what is the importance of this flower? Whenever this flower bloom or open, it produces very pungent smell, just like a carcass, and it attracts many type of flies, then moths, and then small animals and snakes. So see, these organisms come and visit this flower in order to get some reward, and some organisms they lay their egg in the flower. So when the environment is favorable, this egg hatches and produces the young ones inside the flower. Okay, so this is how this plant gets the help of animals or flies to get pollinated. So same thing is given here. It may grow up to 6 feet flower. Okay, a small relationship exists between the species of moth and the plant yucca. This I will show you in the next picture. Here you can see this is yucca plant. Okay, here you can see the shape of the leaf. In garden they usually grow this plant. This is the very big inflorescence. Each one is a flower. Now here I have shown a part of the flower where you can observe the stigma and pollen grains. This is the moth. So there is a coexistence between this moth and the yucca plant. What it will do, moth visit the flower, undertake the pollination, and they lay their egg. So there is an interaction. This egg hatch inside the flower, in the sense when it undergo pollination. The seeds will make sure that growing this moth will get sufficient nutrient. At the same time, flower get pollinated. Okay, so those interactions are very important for the competitive exams. Remember, okay. Then outbreeding devices. This is again a very, very important concept. What is outbreeding device actually? See, most of the plants they show self-pollination, because self-pollination assures that fertilization takes place and plant can produce the seed, and seed will germinate and produces the next generation. But what happens when continuous self-pollination takes place? There is a high chance that inbreeding depression will take place. In the sense, for over a period of time, because of same genes, the offsprings may not be able to adjust to the changing environmental conditions, so that plant may undergo extinction. So majority of the plants, they try to encourage cross-pollination rather than having self-pollination. Even though they are bisexual, they encourage cross-pollination over self-pollination. So this outbreeding devices are the tools through which plants they achieve cross-pollination over self-pollination. We will study one by one what and all the types of outbreeding devices are present in plants. So hermaphrodite flower means what? It has both androecium and gynoecium, okay, the bisexual flower. Continued self-pollination results in inbreeding depression. Even in animals also we can see this inbreeding depression. So what happens? Sometimes continuous self-pollination, at one point of time lethal gene may express or unwanted gene may express, and that particular offsprings are not able to survive in that environment. So in order to avoid that, what animals or plants do? They encourage the cross-pollination. So flowering plants have developed many devices to discourage self-pollination and to encourage the cross-pollination. Those are, in some species, pollen release and stigma receptivity are not synchronized. That means, imagine there is a bisexual flower. It produces androecium as well as gynoecium, but the maturity of pollen grains and the receptivity of stigma are not synchronized. So when the stigma get receptive, pollen grains are not even matured. So what happens? The nearby same species plant, when it produces mature pollen grains, with the help of wind or insects, come and deposit it on the stigmatic surface and cross-pollination takes place. Even though plants are bisexual, here in the case of this condition, we call it as protandry or protogyny. Either androecium is mature first or gynoecium mature first. Okay, so these are the technical terms not given, but remember, protandry means androecium matures first, protogyny means gynoecium matures first. Okay, so with the help of a slight change in the maturation of gynoecium and androecium, these plants they achieve the cross-pollination. Either the pollen is released before the stigma becomes receptive, or the stigma becomes receptive much before the release of pollen. So this is how cross-pollination is achieved. Some other species, the anther and stigma are placed different positions so that pollen cannot come in contact with the stigma. So this is the positional change. Imagine stigma reaches a very height and pollen grains are produced at the base. Now these pollen grains cannot be deposited on the stigmatic surface or vice versa. Anther is long and stigma is short, and in between there will be a physical barrier. Then pollen grains cannot dust on the stigmatic surface. So this is how they discourage self-pollination and encourages the cross-pollination. Both these devices prevent autogamy. So just recollect the kinds of pollination. Autogamy means what? It is a self-pollinated flower where there is a guarantee that pollen of the same plant come in contact with the stigma of the same flower, but that will not happen here. Autogamy will not take place. Again, this is very important concept in theory, in CET and in NEET. Why? Because now they are connecting this topic with the kinds of pollination. The third device to prevent the inbreeding is self-incompatibility. Self-incompatibility means bisexual flower produces the pollen grains and two viable pollen grains, but this viable pollen grains unable to germinate on the stigma because there will be certain chemicals which will detect the self pollen and will not allow this pollen to germinate on the stigmatic surface. So the pollen from another plant of same species can germinate on this stigma. This condition we call it as self-incompatibility. So this is the genetic mechanism, very important. This is the genetic mechanism. In all these outbreeding devices, which one is the genetic mechanism actually? Self-incompatibility is the genetic mechanism and prevents self-pollen from the same flower or other flowers of the same plant from fertilizing the ovule by inhibiting pollen germination or pollen tube growth in the pistil. As I explained, there will be a genetic mechanism which will detect the self pollen from the same flower or the different flowers of the same plant. By that, it will completely avoid autogamy as well as gynoecum. Another device to prevent the self-pollination is the production of unisexual flowers. So obviously when there is a unisexual flowers, the plant has to depend completely on cross-pollination. So in the cross-pollination, there is high chance that pollen grains will be carried by the insects or any type of birds or any type of wind. So this pollen grain is from the different plant belonging to same species. Okay. So in several species such as papaya, male and female flowers are present on the different plants. So this is one best example for dioecious plant. Okay. So that is each plant is either male or female. You can correlate this particular example with the date also. In date plants also you can observe male plant and female plant. So there is a high chance that these flowers undergo cross-pollination. This condition prevents both autogamy and gynoecum. As I said, autogamy means a self-pollen will pollinating the flower. Gynoecum means pollen from the different flower of the same plant pollinating the flower. Both will not take place in this case. So one is self-incompatibility. Second thing is unisexual flowers. So far we studied 4 different types under outbreeding devices. The first one is protandry and protogyny. Second thing, heterostyly. That means the length of the anther and the filament and length of the style is different. And the third one is self-incompatibility, and the fourth one is unisexuality. Remember these 4 words. Keep these 4 words in your mind. It's very, very, very important topic. Okay. So what is the most important aspect of outbreeding device? To encourage cross-pollination over self-pollination. Pollen pistil interaction. So far what we have studied is that is something related to pollination, right? Now pollination is a process where anther is now transferred, pollen grain is transferred from anther to the surface of the stigma. Now the question is, what happens when the pollen grain lands on the stigmatic surface? That process we will study in pollen pistil interaction. There are some pictures given here. Imagine hundreds of pollen grains, they deposit on the stigmatic surface. See, in second PUC in lab we will study some experiments where we will collect the stigma from certain plants, and those stigma are boiled, and with the help of cotton blue stain they are squashed, and those stigma are observed under the compound microscope. What you can see on the surface of the stigma, there are lot of hair-like structure and hundreds of pollen grains. Some of them they germinated and pollen tubes are clearly observed in the style. So this is how we can observe the germination of pollen grains on the stigmatic surface. Now the question is, how this pollen grain will undergo pollen tube formation, or it undergo germination? There are certain type of chemicals which allow the pollen grains to germinate, which is somewhat similar to 10% sucrose solution. In laboratory we use 10% sucrose solution to germinate the pollen grain and observe the pollen tube formation from the germ pole. Here on the stigmatic surface, again somewhat similar to this sucrose solution will help the pollen grains to germinate. First and the foremost thing, the pollination will not ensure the fertilization, because the pollen grain may come from any plant. Pollen grain may not be come from the same plant and land on the stigmatic surface. So the first thing need to be done is to find out whether the pollen grain belongs to same species or different species. So there are some genetic mechanism, with the help of that plant will detect the pollen from the same plant or same species, then only it allows the pollen grains to germinate. Hundreds of pollen grains are deposited on the stigmatic surface, and they starts to produce the pollen tube, and the pollen grain which have the ability to produce the pollen tube faster reaches the ovary where ovules are present. Now pollen tube come in contact with the micropylar region of the ovule and releases its male gametes. Now question comes, how many male gametes are present? Already we studied that male gametes are 2. But sometimes what happens, in 60% of the plants pollen grains are shed at the 2-celled stage. Now when they are released at the 2-celled stage, one of the nucleus which is present in the vegetative cell help in guiding the pollen tube formation, and one of the nucleus which is present in the generative cell acts as the male gamete. Now this generative cell, while moving along with this pollen tube, in the half the way they undergo mitosis and produces another male gamete. So when the time it reaches the micropylar region, there will be 2 male gametes. Okay, rest of the 40% of the plants, they already produce the 2 male gametes before the shed. So pollen grains are released in 3-celled stage in those 40% of the plants. Okay, so same thing is explained here. First they will plant stigma will find out whether it is a compatible pollen or incompatible pollen, and then allow it to germinate. Okay, so when pollen tubes are formed, they will pass through the tissue of the style and then reaches the ovule. Here you can see this is the pollen tube. Now it reached the micropylar region. Here the embryo sac is present. Now embryo sac is consisting of egg apparatus, polar nuclei and antipodal cells. Okay, so the region where the pollen tube enters, that is the micropylar region, and the opposite region is the chalaza. So let us see what and all the explanation is given here. Pollination does not guarantee the transfer of right type of pollen. Why they are saying this? Because the insect which is visiting one flower, you cannot say it visit only one species. So it has visited several species. Its body is having different pollen grains. So there is high chance that the pollen of desirable plant may deposit on the stigmatic surface. Often pollen of the wrong type, either from other species or from the same plant, also land on the stigma. Okay, so there is a reason to give if it is self-incompatible, because if the plant is encouraging outbreeding device or cross-pollination, in that case, suppose insect is carrying the pollen from the same flower, is also a problem for the plant. So at that time, pistil has the ability to recognize the pollen, whether it is of the right type, or you can say compatible type, or the wrong type. If the right type, if it is right type, the pistil accept the pollen and promote the post-pollination events that ends to fertilization. If the pollen is of the wrong type, the pistil rejects the pollen by preventing the pollen germination on the stigma or the pollen tube growth in the style. The wrong pollen grains are deposited on the surface of the stigma, then they are rejected. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of continuous dialogue between the pollen grain and the pistil. See how beautiful it is. We think we are brilliant. Just look at the plants. They have ability to recognize the pollen grains just on their surface, right? Yes. Now the dialogue is mediated by chemical components of the pollen interacting with those of the pistil. So it's the only recent years that botanists have been able to identify some of the pollen and pistil components and the interactions leading to the recognition followed by acceptance or rejection. So far, we tried hard to understand what type of interaction takes place between pollen grain and the stigmatic surface. Recently, scientists, they are able to find out the type of interaction taking place between pollen grain and the stigmatic surface. The pollen grains germinate on the stigma to produce pollen tube through one of the germ pore. One of the germ pore means what? Please do remember with this sentence, one pollen grain does not have one germ pore. They have multiple germ pores. Okay, but from the one germ pore which is directly facing the stigmatic surface, it starts to produce the pollen tube. The contents of the pollen grain move into the pollen tube, and the pollen tube grow through the tissues of the stigma and style and reaches the ovary. Then pollen grains are shed at the two cell stage. It is just for recall they have given. Already I explained what happens when it is released in 2 cell stage and when it is released in 3 cell stage. When it is released in 2 cell stage, when it travels through the pollen tube, in the half the way it again undergo mitosis and produces the 2 male gametes. When the pollen grains are released in the 3 cell stage, 2 male gametes just follow the pollen tube and reaches the embryo sac. Now pollen tube, after reaching the ovary, enters the ovule through the micropyle and enters the synergids through the filiform apparatus. This is very, very interesting actually. Just observe what happens when pollen tube reaches the micropylar region. The nucleus which is guiding this pollen tube reaches micropylar region and its function is over. Now there are 2 male gametes. Somebody has to guide them and help them to fuse with the egg cell and the polar nuclei. That function is done by filiform apparatus. Okay, so these synergids lie near to the egg cell, they have finger-like projections. These finger-like projections guide this male gametes, male nucleus towards the egg cell and polar nuclei. Now what happens? One of the male gamete fuses with the egg and forms syngamy. This is very important process, nothing but the fertilization. Now one more male nucleus fuses with the polar nuclei and forms the triple fusion. One is syngamy, another one is triple fusion. Here you observe two fertilization. One is fertilization of egg, another one is fertilization of polar nuclei. So the term is used double fertilization. And one more term is used, that is triple fusion, because polar nuclei is already diploid. Now it is fusing with the haploid male nucleus. The process is called triple fusion. So same thing is explained here. See, one of the experiment which we perform in the laboratory, that is given here. Take a pollen grain from different plants or any one of the plant, say for example impatiens balsam or any of the nearby plant. You take the pollen grains like hibiscus. Then take the pollen grain of a very young flower, that means the flower which bloom in the morning, and dust this pollen grain on the slide having 10% sucrose solution. Keep this slide for 20 to 30 minutes for observation. Then observe the slide. You see the pollen grains forming pollen tubes. Okay, so there are multiple germ pores. A small pollen tube will be formed in those areas, and one of the germ pore produces very long pollen tube. Okay. Now coming to one of the very important technique. See, artificial hybridization is one of the very important technique practiced long back to produce the hybrid plants. See, how to assemble desirable characters to get the best offsprings. Scientists, they search different ways and they come up with the idea. By knowing the knowledge of pollen-pistil interaction, they just implemented those techniques in artificial hybridization. Suppose the plant is bisexual, produces androecium and gynoecium. A plant breeder, what he will do? He will take the plant, say for example pea plant, before the anthesis, before the flower opens, he take out the petals and he observe the stamens. With the help of a suction pump, a small suction pump, or with the help of needles, he remove all the male reproductive part, that is stamen. This process is called emasculation. Very important. In artificial hybridization, emasculation means what? Removal of stamen from the flower. Now once the stamens are removed, you are very sure that the self pollen cannot deposit on the stigmatic surface. Now in order to make sure that the pollen from some other plant come and deposit on the stigmatic surface, what he will do? He will cover the flower with the help of very thin transparent polythene bag or any type of muslin cloth. Now in the bagging process, the whole flower is covered. Next what he will do? He will take the pollen grains from the desirable plant. Somewhere he observed one plant, it is producing very high yield with the disease resistance. So he collected those plant pollen grains, and here what he is growing is the type of plant which has drought resistance. So he want to include those two characters. So he dusts those pollen grains on the stigmatic surface when the flower matures. Now this we call it as artificial hybridization technique. Artificially you transfer the pollen grain, and rebagging is done to avoid further contamination of the stigmatic surface. So this makes sure that the desirable pollen is transferred on the stigmatic surface and produced offsprings will be hybrids. Okay, but there is no guarantee that produced offsprings will be hybrid, but there is high chance that produced offsprings will be hybrids. If the female parent produced unisexual flowers, there is no need for emasculation. Okay, suppose the flowers are unisexual, then there is no need of emasculation. Simply you take the pollen grain from the desirable plant and then transfer it to this flowers and rebag it. There is a high chance that you will get the hybrid plants. So this is how artificial hybridization is conducted to get the better plants or better offsprings. Now coming to double fertilization. See, so far we traveled so much with this sexual reproduction in flowering plants. We started from Microsporogenesis, Megasporogenesis, and after that pollination, in that kinds of pollination, then different modes of pollination, in that water pollination, wind pollination, insect pollination. See, after that pollen pistil interaction. All these process is nothing but pre-fertilization events. Now we will study fertilization. What happens when the male gamete come in contact with the female gamete, right? See, this is very interesting part. Already we discussed a little about this double fertilization. As I said, pollen tube, they germinate in the on the stigmatic surface and passes through the style and reaches the ovary, where in the locule so many ovules are present. So many pollen tubes, they reach the different ovules and they release the male gametes in the micropylar region, and these male gametes are directed by the filiform apparatus which is present on the synergids, and they guide these male gametes to reach the egg and polar nuclei. So one of the male gamete fuses with the egg and leads to syngamy. It makes sure that now fusion of male and female gametes leads to the formation of zygote. In other words, at the same time, another male gamete fuses with the polar nuclei, and we call it as triple fusion, and which makes sure that this polar nuclei after fertilization develops into endosperm. There is one important aspect here. Why there is a need of endosperm? And one more thing, endosperm formation takes place first and embryo development takes place later. Sometimes in competitive exam they particularly take these sentences. Okay, so the endosperm formation takes place first and embryo development takes place later. The reason is developing embryo make sure that there is sufficient food for its development. So endosperm development takes place first and then embryo development takes place. Now there are the high chance that the questions may ask based on the ploidy. Now tell me, what is the ploidy of endosperm? It is triploid because primary endosperm nucleus is already diploid. One more male gamete come in contact with it and formed a endosperm. Now the endosperm is 2 plus 1, that is 3. It is triploid. Endosperm is always triploid, and the zygote formed is diploid. Okay, so this process we call it as double fertilization. Fertilization of egg and fertilization of primary endosperm nucleus takes place simultaneously, and this process we call it as double fertilization. Okay. Now here in this picture you can see that the synergids after fertilization, they undergo degeneration. That means whatever the nutrient content is there, that will be consumed by developing zygote, and the endosperm, nourishes the developing zygote, and zygote will develop into embryo. These antipodal cells also undergo degeneration. This zygote later develop into different stages of embryo that we will study in detail in embryo. Here you can see globular embryo, heart-shaped embryo, and a mature embryo where you can clearly observe the cotyledons. So this is definitely an example of dicotyledon. So in this post-fertilization structure and events, we study in detail about the endosperm, in detail about the zygote developing into embryo. So we'll study embryos, and then what happens to ovule. The fertilized ovule develop into seed, we study in detail. What happens to ovary? Ovary develops into fruit, that we will study in detail. Okay, so these are the aspect which we are going to study in post-fertilization structure and events. Let us see one by one. In that, first one is endosperm. Just now I said, what is endosperm actually? When the primary endosperm nucleus undergo fertilization with the help of male gametes, endosperms are formed. Now tell me whether endosperm is formed first or embryo development takes place first. Definitely endosperm formation takes place first. Now whether this endosperm is retained in the seed or it is completely consumed, or which is the form of endosperm we commonly observe. Hope you have tasted a tender coconut. What is this tender coconut actually? It is the free nuclear endosperm. This endosperm is formed continuously by multiplying its nucleus, which is not followed by the cell wall formation. So only after the maturation of coconut you see a kernel which is white colored, where endosperm nucleus is now surrounded by the cell wall formation. Before that it is all a nucleus, free nuclear endosperm. This endosperm sometimes completely consumed by the growing embryo. Such seeds are called non-endospermic seeds. So can you give me some example for non-endospermic seeds where endosperm is completely consumed by growing embryo? The best example is dicotyledons, like groundnut, like badam, like cashew nut, where completely this endosperm is consumed. If you cut open the seed and observe, there will be a groove which is empty, and at the base of that the small embryo is observed. Can you tell me some example where endosperm is retained even after the seed formation? The best example is monocot seeds like wheat, rice, coconut, castor, where you observe the endosperm even after the complete embryo development, and in the seeds these endosperms are present. So based on that we will classify them as albuminous seeds and non-albuminous seeds. Albuminous seeds means endosperm is present, non-albuminous seeds means endosperm is completely consumed. Okay, so that is about the endosperm. Next part is about the embryo. Now the study of embryo is why so important. Whether the embryos are same in all the cases, in the case of angiosperms, no, that's not true. In angiosperm we observe major two classifications, that is monocots and dicots. Now monocots have one type of embryo and dicots have one type of embryo based on their cotyledons and some other structures that we will study in detail. Okay, just observe this picture. What happens to zygote? Zygote is diploid. As it starts to divide mitotically, the number of cells will increase, and initially it just form a mass of cells, and those mass of cells they slowly starts to differentiate, and first it forms the globular embryo. So this is the structure of globular embryo. This is the suspensor actually. This part actually attaches to what it attaches to micropylar region. Okay. Now this suspensor at the end, you can see here it is heart shaped, and finally it completely matures and forms the two cotyledons. Now the same structure is given here. Now when you observe this dicot embryo, where you can differentiate the hypocotyle region and epicotyle region. In the epicotyle region, you observe the plumule part and cotyledons. Actually, this plumule, after germination of the seed, develop into shoot. In the hypocotyle region, you observe the tubular part. At the end, radicle is present. This radicle later develops into root. Okay, this root is covered by a protective layer that is called root cap, which is made up of calyptrogen. So the thing is, dicots, they have two cotyledons. Now my question is, what is this cotyledon? Why they are present in the embryo? And even in the seeds you can see the cotyledons. Whenever you cut open the groundnut seed, you can see two cotyledons. Or any of chana dal or anything, you cut open the seeds, you observe two cotyledons. They are actually leaf of the embryo. Whenever this seed starts to germinate, it doesn't have a proper root system to support the plants, or it needs to develop the root system that requires the food. So those food is stored in the cotyledons, and with the help of cotyledons, first they establish well root system, and then with the help of the nutrients remained in the cotyledons, they produce the shoot system and then produces the leaves. So before the leaf formation, cotyledon acts as an embryonic leaf. Please do remember, this is the very important concept. Okay, hope you have just observed the dicotyledon embryo. Now observe the monocotyledonous embryo. There are a lot of changes in monocotyledonous embryo. Why the name monocotyledon? Because it has only one cotyledon. That one cotyledon is represented by the scutellum. Okay, scutellum represents the single cotyledon. Now if you observe the embryo, this is the shoot part, nothing but shoot apex, you can say plumule, and that is covered by a protective layer called coleoptile. This was not observed in dicotyledonous embryo. Here plumule is covered by coleoptile and radicle is covered by coleorhizae. Okay, here you can see a small part called epiblast. Explanation is not given in textbook, but can you predict what is that epiblast and why it is given? See, it's just a ruminant part. Okay, actually if it grows and form one more cotyledon, it would have observed like a dicotyledonous seed. But in evolution, this part got reduced. Okay, it's a reduced cotyledon. And one cotyledon is very prominent, that we call it as scutellum, that acts as a single cotyledon in the case of monocotyledons. Hope this is very clear. Here shoot is covered by a protective layer called coleoptile and root is covered by a protective layer called coleorhizae. So this is the main difference between monocot embryo and dicot embryo. Next concept is seed. See, tell me, what is seed actually? Embryo is nothing but the zygote which develop and becomes embryo. Where it is present? It is present inside the seed. Now what is seed? Right? Now just recollect the ovule. Inside the ovule, embryo sac is present. Now embryo sac has egg apparatus. Now egg after fertilization develop into embryo. Now what happens to the ovule? That same ovule after fertilization develop into seeds. Okay. Now what is the importance of seeds in plants? Because the production of seeds makes sure that that plant can produce the next generation. Because in lower plants we observed that the body of the plant itself undergo breakdown by fragmentation and each fragment grow into a new plants. Or there are certain mechanisms where the offset, the suckers or different types of structure produced in the vegetative body of the plant itself give rise to a next generation. But in sexual mode of reproduction, this is the mean for production of offsprings, seed. So please do remember that seed is nothing but fertilized ovule. Now, is there a connection between the number of ovules and number of seeds? Of course. The number of ovules get fertilized will develop into number of seeds. Okay. Now in angiosperm, seed is the final product of sexual reproduction and it is often described as a fertilized ovule. Seeds are formed inside the fruit. Now what is fruit actually? Fruit is nothing but the fertilized ovary. Inside that ovules are present, right? So ovules are fertilized and develop into seeds, and ovaries are after fertilization develop into fruits. A seed typically consists of seed coats. How these seed coats are formed? Just observe the ovule. You can see the integuments. These integuments later develop into seed coat. That seed coat, inner seed coat is called testa and outer seed coat is called tegmen. The cotyledons of the embryo are simple structures, generally thick and swollen due to the storage of food reserves. The cotyledons, which is part of the embryo, which is mainly for storage of food. Mature seeds may become non-albuminous or X-albuminous. Why they become non-albuminous or X-albuminous? Because growing embryo, if it completely consumes the endosperm, then we use the term non-albuminous, X-albuminous or non-endospermic. All these terminologies are same. Non-albuminous seeds have no residual endosperm as it completely consumed during the embryo development. There are some exceptions also. Okay, albuminous seeds retain part of the endosperm as it not completely used during the embryo development. Like in the case of wheat, maize, and barley and castor, they retain the endosperm even in the seeds, and those seeds are called albuminous seeds or endospermic seeds. Okay. Occasionally, some seeds such as black pepper, this is one exception. Please do remember, very important example. Black pepper in beet, remnants of the nucellus are also persistent. Okay, the terminology used for this remnant nucellus or persistent nucellus is perisperm. Most of the time in competitive exam, they focus on this type of terminologies. What is the meaning of perisperm? Persistent nucellus. Persistent means what? Actually, after fertilization, nucellar cells should undergo degeneration. But even after the fertilization, in the seeds, you observe nucellar cells, and such persistent nucellus is called perisperm. Now, integuments of the ovule harden as tough protective seed coats. The micropyle remain as a small pore in the seed coat. If you observe any of the seeds, like you take the example of Kabul or Channa, observe its end. At one point there is a hole. That hole is nothing but what? Actually a micropylar region. Why it is present? If you soak the seed in the water, water can enter through the micropylar region and it helps in imbibing the water, and seed can swell, and this swelling help them to germinate. Okay, so the micropylar region will be persistent even in the seeds also. This facilitates the entry of oxygen and water into the seed during germination. As the seed mature, its water content reduced and seed becomes relatively dry to 10 to 15%. Now the question comes, why seed should undergo dry? See, most of the times on set of favorable conditions, plant will grow and they produce very beautiful vegetative structures, and they produce flowers, and flowers undergo pollination, and seed setting will take place. When the time comes to seed setting, the plant will experience a unfavorable condition or harsh condition. In case the produced seeds, when they fall on the soil and immediately undergo germination and produces the plantlets, and plantlet starts to grow, the coming seasons are completely unfavorable, like it may be winter or it may be summer. So there is very less chance that this plant will survive and produces the next generation. So in most of the case, what happens, the produced seed will retain very less amount of water, 10 to 15%, and undergo dormancy. What is dormancy actually? It is a inactive viable time of the seed. Inactive viable means what? Seeds are viable. They have ability to germinate in onset of favorable condition. But for a time interval, it slowed down all its physiological activity and undergone dormancy to make sure that this seed will be viable in the unfavorable condition, and onset of favorable condition they can germinate. Okay, so the general metabolic activity of the embryo slows down, and embryo may enter a state of inactivity called dormancy. This dormancy will vary. Okay, you cannot say seeds may be dormant for 3 months, seeds may be dormant for 4 months, or seeds may be dormant for 1 year. It varies from plant to plant. There are certain examples, you will be surprised. Some seeds are extracted from the place where these seeds are viable for thousands of years. Surprising, right? For thousands of years, they retain their viability. I will give you those examples. Okay, as the ovule mature into seed, the ovary develops into fruit, and the transformation of the ovule into seed and ovary into fruit proceeds simultaneously. It is not a separate process. First seed formation takes place, then fruit formation takes place. There is no such things. So these two, formation of seed as well as formation of fruit, takes place simultaneously. Wall of the ovary develops into a wall of the fruit called pericarp. In morphology, you have studied this. What is pericarp? Pericarp is present external to the endocarp, where mesocarp and epicarp is present. Okay, the fruit may be fleshy, as in guava, orange, mango, etc., or may be dry, as in groundnut and mustard. There are different types of fruits: fleshy fruits, dry dehiscent fruits. Okay, so some of the examples you studied in morphology of flowering plants. Many fruits have evolved mechanism for dispersal of seeds. So this is a really interesting type of mechanism. Have you seen impatiens balsam or balsam plant? If you just touch the mature fruit, the fruit will burst and releases the seeds to the environment. Okay, so the different plants have different techniques. You can see a papaus. During winter season, a small parachute like seed will fly around us. This is also a type of seed dispersal mechanism, parachute mechanism of seed dispersal. Okay, so is there any relationship between number of ovules in the ovary and the number of seeds in the fruit? Of course there is a relationship, because the number of ovules present inside the ovary, same thing after fertilization develop into seeds, and the ovary develops into fruit. Now in most plants, by the time fruit develops from the ovary, the other floral parts degenerate and fall off. Why they have used the term most of the plants? There are certain examples. Let me tell you, in the case of brinjal, tomato, or in capsicum, the sepal will retain on the fruit also. That we call it as persistent sepals, right? So they have used the term most of the plants. Okay. However, in a few species such as apple, strawberry, cashew, the thalamus is also contribute to the fruit formation. Tell me, what if what if other than ovary forming a fruit, what we call it as? True fruit or false fruit? Of course, it's a false fruit. Sometimes what happens, the ovary will not develop into fruit, instead thalamus will develop into fruit. So for that they have given some of the example like apple, strawberry and cashew. These are the best example for false fruits. Such fruits we call it as false fruits. Most fruits however develop only from the ovary and are called true fruits. You have hundreds of different examples for true fruits, where most of the times ovary develops into fruit after fertilization. Although in most of the species fruits are the result of fertilization, there are few in which fruits develop without fertilization. See, this is again an exception. Most of the times the fertilized ovary develops into fruit, but there is a chance where fruits are produced without fertilization, and those fruits are called parthenocarpic fruits. So in nature you observe one of the best example, that is banana. Have you seen banana plant? Now tell me, if the ovary is directly developing into fruit without fertilization, what is the essentiality of producing flowers in banana plant? Stigma, style, petals, everything is just a waste. It just requires gynoecium, and that gynoecium develops into fruit. So that's why in banana you can see it produces very much large colored bracts. They are simply covering all the flowers, because flowers, they do not require any type of pollinators. Even though the insects, if they visit the flowers, they will get reward in the terms of nectars, but they don't require insects for pollination. Fruits are produced without fertilization, and that condition we call it as parthenocarpic fruits. Now the question comes, when the fruits are produced without seeds, it's very easy to eat. Can we induce this character in any of other plants so that we can produce a type of without seeds? Imagine watermelon without seeds. Imagine grapes without seeds. Imagine orange without seeds. How easy to eat, right? So there are some practical applications where they use plant growth regulators like auxin to produce parthenocarpic fruits. So in market you will get seedless grapes, seedless orange. All these are what? Actually they used plant growth regulator like auxin to produce the seedless parthenocarpic fruits. Here are some examples. You can see this is the thalamus in the case of apple, and this is one monocot seed vertical section. Here you can see the endosperm. So the maize seed is an example for endospermic seed. This is the scutellum, single cotyledon, and here you can see coleoptile, plumule, radicle and coleorhizae. What is this pericarp actually? Proteinaceous layer, that is pericarp. Okay, so here is the strawberry. These are some of the example of embryo, and this is the cotyledon in the case of dicots. There are some questions which appeared from the these diagram based questions in NEET and CET, that we will discuss in the later part of the topic. As they have sufficient food reserve, young seedlings are nourished until they are capable of photosynthesis on their own. That's why earlier in this topic I explained what is the role of cotyledons. They act as embryonic leaf. Okay, the hard seed coat provides protection to the young embryo. The being products of sexual reproduction, they generate new genetic combinations leading to variation. What is the main intention of sexual mode of reproduction? Producing offsprings having some variations. So what is this variation and why it is so important? Variation leads to a better chance to the offsprings to survive in the environment. So seed is the basis of our agriculture. So in agriculture, seed is the basic thing. Farmers used to grow the plants. At the time of harvest, they use some amount of seeds for commercial purpose, some amount of seeds for their personal use, that means as a source of food, and some amount of seeds will be stored for the next year to produce a new crop. Okay. Dehydration and dormancy of mature seeds are crucial for storage of seeds which can be used as a food throughout the year and also raise crops in the next generation. Imagine if the plants are all like tomato. In the case of tomato, you cannot dehydrate the seeds. If you want to grow the tomato, you just have to cut open the tomato fruit and place the seeds directly in the soil. It will grow into a new plant. You cannot dehydrate it. It requires lot of moisture. But in some plants like rice, they only retain 10 to 15% of moisture, so which makes sure that they will survive in the unfavorable condition, and on set of favorable condition they can be used for the production of new crops. So can you imagine agriculture in the absence of seeds? We cannot imagine, but there are possibilities. Okay. Presence of seeds which germinate straight away soon after formation cannot be stored. How long do the seeds remain alive after they are dispersed? This period again varies greatly. That means in plant to plant, this condition, that means dormancy, viability, dehydration will be from plant to plant. There are some examples here. The oldest is that the Lupinus arcticus, excavated from Arctic tundra. Okay, so this plant, Lupinus arcticus, seeds germinated and flowered after estimated of 10,000 years. Surprising, really surprising. Okay, this Lupinus arcticus plant seed retained its dormancy and viability for 10,000 years. Now tell me whether tomato seed can be stored this amount of or this duration of time. It is not possible. So it varies from plant to plant. One more example, there is a recent record of 2,000-year-old viable seed is of the date palm, that is Phoenix dactylifera, discovered during the archaeological excavation at King Harold's palace near the Dead Sea. Again they got one seed of date palm which is around 2,000 year old and it is viable. They produced the plant from those seeds. So these are the two extreme examples where seed can be viable even up to 10,000 years. Now in NCERT textbook they asked some of the questions. We will try to answer those questions. Okay. How many eggs are present in an embryo sac? How many eggs are present? Only single egg. How many embryo sac are present in an ovule? Single embryo sac. How many ovules are present in an ovary? This will vary. It will vary greatly from plant to plant. How many ovaries are present in a typical flower? This will also vary. Sometimes monocarpellary, bicarpellary, tricarpellary, some multicarpellary conditions are observed. Then how many flowers are present in a tree? Sometimes we observe a tree with full of flowers, thousands of flowers. Sometimes a tree is producing single flower in its lifetime. So like that, there are so many variations we observe in the number of flowers. Now some more exceptions or interesting characters are given. Orchid fruits are one such category, and each fruit contains thousands of tiny seeds. That means if you observe orchid gynoecium or ovary, how many ovules may present? Thousands of ovules are present. After fertilization, all those ovules develop into seeds. That's why in the fruits we observe thousands of seeds. If possible, you just observe the vanilla pod. Inside that vanilla pod, thousands of seeds, tiny seeds are present. Okay. There are two more examples, that is orobrac and striga. Here I have given the orobrac and striga. You can see the plants. If you observe their fruits, inside that it produces thousands of seeds. They are parasitic plants. Okay, so in examination they may ask this example as a parasitic plant. Have you seen tiny seeds of ficus? Ficus plant, have you seen? In that it produces so many fruits, and frugivorous birds, they used to come and visit the plants at the time of fruiting. And if you cut open that fruit and observe, seeds are very minute. Okay, but thousands of seeds are present. Then how large is the tree of ficus developed from that tiny seed? That is the beauty of nature. See, that tiny seed has genetic information. It can produce a tree which is of a size of an acre, right? So how many billions of seed does each ficus tree produce? Even though they produce billions of seeds, possibility of producing offsprings will be very, 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 very less. From that billion seeds, it can produce one or two offsprings. Okay, can you imagine any other example in which such tiny structures can produce such a large biomass over the years? They are asking some other example which is similar to ficus plant. Okay, if you want, you can search such type of plants in nature. Now the last part of the topic, and very interesting part of the topic, apomixis and polyembryony. So far we studied that even without the fertilization, ovary can develop into fruit, and that process we call it as parthenocarpy. Now just observe here, although seeds in general are the products of fertilization, a few flowering plants such as some species of Asteraceae and grass have developed a special mechanism to produce seeds without fertilization called apomixis. This is very, very important example. In the case of Asteraceae and grass, seeds are produced without fertilization. We studied production of fruits without fertilization, but here seeds are produced without fertilization, and that process we call it as apomixis. Now tell me, if the seeds are produced without fertilization, is this an advantageous process for us? Maybe for plants, some of the plants it is a type of adaptation for survival. For us it's really a boon, because apomictic seeds, if they are available, they are just like clones. The offspring produced are exactly similar to their parents. So scientists are very curious about how these apomictic seeds are produced in Asteraceae family and in grass. They are trying to introduce these apomictic seed production in other plants also, so that any farmer can get benefit from it. There is no need to visit the seed bank or any of the commercial scale production of seeds every year to grow the plants. Other than that, he can collect the seed from his own field and retain its hybrid character for many generations. Actually, what happens, once he collects the seed from the hybrid seed banks and sow it in the field, because of cross pollination, all the genetic compositions will be changed, and in the next generation, if we take the seeds from the same plant and sow it in the field, there is a high chance that he loses hybrid vigor. So introduction of apomictic seeds in the agriculture may be profitable for farmers. They can use the seeds produced in their own farm and use it for next generation production. Okay, so it has a great agricultural application importance. The fruit production without fertilization is called parthenocarpy. Apomixis is a form of asexual reproduction that mimics the sexual reproduction. Such a beautiful word. Apomixis It's a type of asexual reproduction. Actually, seeds are produced without fertilization, right? It is exactly like a clone, but still which mimics the sexual mode of reproduction. It produces the seed. Seed production is only observed in sexual mode of reproduction. So there are several ways of development of apomictic seeds. In some species, diploid egg cell is formed without reduction, division, and develops into an embryo without fertilization. Okay, so more often as in many citrus and mango varieties, some of the nucellar cells surrounding the embryo sac start dividing, protruding into an embryo sac and develops into embryo, and such species each ovule containing many embryos, and that is called polyembryony. What happens here actually, inside the ovule, one embryo sac will be there, one egg will be there. After fertilization, zygote will be formed, zygote will develop into embryo. But here in this case of citrus and mango, what happens? Nucellar cells also becomes embryos. Okay, that means from one seed you can observe multiple embryos. That condition is called polyembryony. And occurrence of more than one embryo in the seed is referred to as polyembryony. What would be the genetic nature of apomictic embryos? Genetic nature, they are clones. Can they be called clones? Of course, yes, they are clones. Now, what is its application in agriculture? We'll try to understand. Hybrid varieties of several of our food and vegetable crops are being extensively cultivated, and cultivation of hybrids has tremendously increased the productivity. When the productivity is very high, usually farmers, what they will do, every time they go to a shop where they sell the hybrid seeds. So the cost of the hybrid seeds will be very high. See, in agriculture, if they spend more on hybrid seeds or chemical fertilizers or for labors, then the profit what they get at the end will be very, very less. So if this continues, most of the farmers, they lose their interest in agriculture, right? So in order to overcome this problem, one of the problem of hybrid is that hybrid seeds have to be produced every year. Because of cross-pollination, in the next year itself it loses its hybrid vigor. If the seed collected from the hybrids are sown, the plant in the progeny will segregate and do not maintain the hybrid character. Because of segregation, what happens? The hybrid character cannot be retained. Production of hybrid seeds is costly, and hence the cost of hybrid seed become too expensive for farmers, as I said. If these hybrids are made into a apomictic, there is no segregation of character in the hybrid progeny, then the farmers can keep on using the hybrid seeds to raise the crops year after year, and he does not have to buy hybrid seeds every year. So this will be a great advantage for the farmers. No need to depend on hybrid seeds. They can use the seeds produced in their own crop field. Okay, because of this importance of Apomixis, the hybrid seed industry, active research is going on in many laboratories around the world to understand the genetics of Apomixis and to transfer apomictic genes into the hybrid varieties. If this got achieved, then the farmers will be rewarded better. Their profit ratio will be increased. Okay, so with this we completed the explanation part of the topic sexual reproduction in flowering plants. We studied in detail about the pre-fertilization structure and events, then fertilization and post-fertilization and structure and events, under that endosperm, embryo, seed, fruit, and some of the exceptions like apomixis and polyembryony. Let us discuss some of the questions which appeared in the competitive exam like NEET. Okay, let us continue with the previous year questions asked in NEET examination. So the purpose of explaining this previous year NEET question is, whenever we are studying any topic, we should be very clear in our mind that we are preparing for examination. So whenever we look into the different types of previous year questions, we will have a clear idea what type of approach we need to take to prepare for the competitive exams. Okay, so this is the question which is asked in 2025 NEET. Look at the pattern of the question paper. Here they have asked assertion reason type of question. Given below are two statements. One is labeled as assertion and other is labeled as reason. Assertion given is a typical unfertilized angiosperm embryo sac at maturity is 8 nucleate and 7 celled. Is this statement is correct? Just recollect the embryo sac formation where once the meiosis takes place from the megaspore mother cell, now one of the cell which is nearer to the micropylar region will be retained, and that will undergo 3 successive mitosis and produces 8 nucleus. Out of that 8 nucleus, 3 will move towards one polar region, another 3 will move towards another polar region, and near the chalazal end they form antipodal cells. Near the micropylar end, 2 cells will become synergid, one will become an egg, and that we call it as egg apparatus, and 2 nucleus at the center will form a central cell. That means number of cells will be 7. So the first statement is correct. Now reason, the egg apparatus has 2 polar nuclei. This is wrong. The 2 polar nuclei is present in central cell. That's why the answer is assertion is true but reason is false. Now one more question which is asked in 2025. Both wind and water pollinated flowers are not very colorful and do not produce nectars. Of course, when abiotic factors are responsible for pollination, obviously there is no need of producing colorful petals and nectars or any type of floral reward. Reason, the flowers produce enormous amount of pollen grains in the wild and water pollinated flowers. Now just observe, is this a reason for this? Probably the answer is assertion and reason both are true, and R is not the correct explanation of A. That means these two statements are correct actually. But this statement, the flower produces enormous amount of pollen grain in wind and water pollinated flowers, will not explaining why these plants are producing colorless flowers or small flowers, right? So both the statements are correct, but reason is not the correct explanation of assertion. So another question from 2025. How many meiotic and mitotic divisions need to occur for development of mature female gametophyte from the megaspore mother cell in an angiosperm plant? One meiosis is important. Then it is followed by, in the previous question I already explained, 3 mitotic division and forms the embryo sac. So the answer is 1 meiosis and 3 mitosis. One more assertion reason from the same topic in 2025. Cell of the tapetum possess dense cytoplasm and generally have more than 1 nucleus. This is correct. The presence of more than 1 nucleus in the tapetum increases the efficiency of nourishing the developing microspore mother cell. This is wrong actually. They nourishes the developing pollen grains. So assertion is correct, reason is incorrect. So these are the questions asked in 2025. Just look at the pattern, okay? Just observe the pattern as we move from 25, 24, 23, 22, 20 and 19. You will have a clear idea that nowadays the questions are framed in a way that students need more time to solve the questions. So even though they have clarity about the topic, the speed of solving problem plays a vital role in competitive examination. Okay, identify the set of correct statements. See, there are four statements. It takes time to read it, and then you need time to analyze it. Now here he is trying to kill your time. The flower of Vallisneria are colorful and produce nectar. Absolutely wrong statement, because it is a water pollinated plant. The flowers of water lily are not pollinated by water. That is correct. The most of the water pollinated species, the pollen grains are protected from wetting because of mucilage. That is also correct. Pollen grains of some hydrophytes are long and ribbon like. This is also correct. In some hydrophytes, the pollen grains are carried passively inside the water. This is also correct. Now tell me which one is correct. B, C, D and E only. A is incorrect statement. Now just look at this diagram. See, there is a high chance that in competitive exam they pick up the diagram to frame the question, because what happens most of the time, we read all the theoretical parts properly and we memorize it, but when it comes to the diagrams we used to ignore it. So they found out a way in which they started framing questions on diagrams, so that these students who are neglecting this diagrams, they get very much frustrated when they look at the diagram based questions in examination. Okay, so this diagram, do you remember it is present in which topic? We studied actually corn cob, something related to maize plant. Identify the correct description about the given figure. Water pollinated flowers showing stamens with mucilaginous covering. Absolutely wrong. Cleistogamous flowers showing autogamy. No. Compact inflorescence showing complete autogamy. No. Wind pollinated plant inflorescence showing flowers with well exposed stamens. This is correct. Here you can see well exposed stamens, which helps in dispersal of pollen grains in the wind. Okay, so the fourth answer is correct. Now the junction between ovule and funicle is represented by, it's a very simple question. Junction between funicle and ovule is called hilum. Here the fourth option is hilum. Which of the following represents the female gametophyte? Female gametophyte, not female gamete. If it is female gamete, then the answer is egg. Here they have asked female gametophyte, so the answer is embryo sac. The flower of Amorphophilus provides safe place for pollinators to lay the eggs. This is an example of amensalism, floral reward, anemophily and commensalism. So what type of answer we can include in this topic? Because amensalism, anemophily and commensalism, it is all related to ecology. Now here you can say it is a floral reward. Because of that floral reward, the flies or animals, they visit the flower and undertake pollination. Which part of the ovule store reserve food material? Nucellus, integuments, placenta, funicle. It's a very simple question. Again, answer is nucellus. Pollen grains remain preserved as fossils due to the presence of epidermal layer, tapetum, exine layer, intine layer. This we studied in detail. Actually, exine is made up of sporopollenin, which is indestructible. Because of that reason, pollen can be preserved as a fossils. See the intensity of questions asked in every year. Every year they are picking around 4 to 5 questions for examiner in question papers. Okay. In 2023, what is the function of ears in corn cob? Protect seeds, trap pollen grains, and attract insect to disperse the pollen grains. See, the main function is to trap the pollen grains. If you observe the corn cob, many hair-like structures will come out. What it represents? Actually a style and stigma. These are mainly coming out to trap the pollen grains, airborne pollen grains. Okay. So the answer is 3. In angiosperm, the haploid, diploid and triploid structure of the fertilized embryos have sequentially are. Very simple question. Which one is haploid here? Synergid is haploid. Which one is diploid? Primary endosperm nucleus is here diploid is zygote and triploid is primary endosperm nucleus. The answer is 4th one. Large, colorful, fragrant flower with nectar are seen in wind pollinated plants, bird pollinated plants, insect pollinated plants and bat pollinated plants. Question is very direct. Large, colorful, fragrant flowers. See, when do we observe fragrant flowers? Actually, in case of insect pollinated plants, because birds, they require some kind of reward like nectars. Okay, so the insects are easily attracted when there is a fragrance. In which of the following sets of families the pollen grains are viable for months? So this is nothing but Leguminosae, Solanaceae and Rosaceae, option 4. Identify the incorrect statement related to pollination. So these are the direct questions. Moths and butterflies are the most dominant pollinating agents among the insects. Wrong, it is honeybees. Pollination by water is quite rare in flowering plants. This is correct. Pollination by wind is more common amongst the abiotic pollination. This is also correct. Flowers produce foul odor to attract the flies and beetles to get the pollination. Yes. So the first one is incorrect. There are two statements given here. One, cleistogamous flowers are invariably autogamous. Why it is invariably autogamous? There is no chance that cross-pollination takes place because flower will never open in the case of cleistogamous flowers. Statement 2, cleistogamy is disadvantageous as there is no chance of cross-pollination. So here both the statements are seems to be correct. Statement 1 and statement 2 both are correct. Okay. Which part of the fruit labeled in the figure makes it false fruit? If you observe here, D represents the seed and B represents seed coat, and here it represents the true fruit, and C represents the thalamus. In apple, actually thalamus acts as a fruit. So here you can say C, thalamus acts as a fruit here. So that is the 4th option. What will be the ploidy of endosperm of seed produced after crossing tetraploid female plant with the tetraploid male plant? See, this is like some kind of problem based question. Let us see. Crossing a tetraploid female plant, let us say it is 4N, tetraploid male plant. Okay, so what will the ploidy of endosperm? You should find out the ploidy of endosperm. Now in female, primary endosperm nucleus will be 4N only, because in diploid plants primary endosperm is diploid. That means in tetraploid plant primary endosperm will be tetraploid. So what will be the ploidy of gametes here? In diploid it is haploid, in tetraploid it is diploid. Now 4 plus 2N, how much it is? It is 6N. So the answer is hexaploid. Answer is hexaploid. Okay. Yes, answer is hexaploid. Now, which of the following set represents the correct labeling of A, B, C, D with respect to the given diagram? So again a diagram based question. Here the answer is A represents seed coat, B represents cotyledon, and C represents the endosperm, D represents the hypocotyle. In general, the egg apparatus of embryo sac in angiosperm consists of, what it consists of? 2 synergids and 1 egg cell. Answer is 1. To ensure that only the desired pollen fall on the stigma in artificial hybridization, the female flower buds of the plant producing unisexual flowers need not be bagged. Let us see all the statements and then come to the conclusion. There is no need to emasculate the unisexual flowers of selected female parent. Unisexual female parent, this is correct. Emasculated flowers are to be bagged immediately after cross-pollination. This is also correct. Emasculated flowers are to be bagged after removal of anthers. Correct. Bisexual flowers showing protogyny are never selected for cross. Is it correct? Bisexual flowers showing protogyny. Let us see. So the answer shows B, C and D are correct. E is also a wrong statement. First one and second, last one are the incorrect statements. Remaining are correct statements. Okay. Why? Because female flower buds of the plant producing unisexual flowers need not be bagged. They have given it has to be bagged because you are avoiding the contamination of the stigma by any of the pollen grains except the pollen grain which has desirable character. You collect it to transfer. Remaining all the pollen grains should not enter the stigmatic surface. This is correct. Incorrect statement. Bisexual flowers showing protogyny are never selected for cross. It's not like it should not be taken, because if it is a desirable plant, you can take it for the procedure. Next, the residual persistent part which forms the perisperm in the seed of the beet is calyx, endosperm, nucellus, integument. Perisperm is formed by the persistent nucellus. Again a direct question. Which of the following can be expected if the scientists succeed in introducing apomictic gene into hybrid varieties of the crops? Again last part and very simple. So what you can expect here? Polyembryony will be seen and each seed will produce many plantlets. No. Seeds of hybrid plants will show longer dormancy. No. Farmers can keep on using the seeds produced by the hybrids to raise the new crops year after year. Yes, that is the main reason. There will be a segregation of desired character only in the progeny. No, there will not be any segregation. Offsprings are exactly similar to their parents. They are the clones. So the answer 3 is correct. A typical angiosperm embryo sac is 8, again it is a repetition, 7-celled, 8 nucleated condition. 8 nucleate, 7 option 3. The term used for transfer of pollen grains from anther of one plant to the stigma of the different plant, which during pollination brings genetically different pollen grains into stigma, is chasmogamy, xenogamy, cleistogamy and gytonogamy. It's very easy question. Which will bring the genetically different one, that is xenogamy. There are only 3 types: autogamy, gytonogamy and xenogamy. In first two case they are almost same like self-pollination. Even though the cross-pollination takes place in gytonogamy, it is similar to self-pollination. Only one type where genetically different pollen grains will be brought, that is xenogamy. Answer is 3. In some members of which of the following pairs of families pollen grains retain their viability for months? Again, it is a repeated question. That is Leguminosae and Rosaceae. Option 2. The plant part which consists of two generations, one with the other, are pollen grains inside the anther, correct. Germinated pollen grains with the two male gametes, no. Seed inside the fruit, no. Embryo sac inside the ovule. So option A and D. Option 3. Okay. Which of the following is incorrect for wind pollinated plants? Well exposed stamen and stigma, correct. Many ovules in each ovary, wrong. One ovule in one ovary. Flowers are small and not brightly colored, correct. Pollen grains are not light and non-sticky. So all these are the characters of wind pollinated plants. So the second one is incorrect. Next one. What is the fate of male gametes discharged in the synergids? One fuses with the egg and other fuses with the central cell nuclei. So the first option itself is correct. Which one of the following statement regarding post-fertilization development in flowering plant is incorrect? Post-fertilization event, just recollect. Ovules develop into embryo sac, wrong. Ovary develops into fruit, correct. Zygote develops into an embryo, correct. Central cell develops into an endosperm, correct. So the first one is incorrect. Which is the most common type of embryo sac in angiosperms? Monosporic with the 3 sequential mitotic division. Second option is correct. Direct question again. What type of pollination takes place in Vallisneria? So here flowers emerge above the surface of water and pollination occurs by insect, wrong. Pollination occurs in submerged condition by water, wrong. Flowers emerge above the water surface and pollen is carried by the wind, wrong. Male flowers are carried by water current and female flowers at the surface of the water, correct. So the fourth option is correct. So with this we completed the complete explanation part of sexual reproduction in flowering plants and all the questions which appeared in NEET examination. Hope this video helped you to prepare for your examination. Thank you.